I can see just 3 persons here yet. So let's wait for others to join. Okay, 4 persons. 4 persons. If you're here, just drop your name on the comment section. Just drop your name on the comment section. Now, just like I said, today I'll be doing first come, first serve. So drop your name in the comment section. Just your name in the comment section. The first name I see, the first person I'll attend to. Okay. 5 persons here. It's 5 persons. So we're starting in few minutes time, just few minutes. Let's just wait for others to join. But while we wait for others to join, if you're just joining, just as soon as you join, just drop your name as soon as you join. Just drop your name as soon as you join. All right. Okay, so the first person, Daniel, okay. Okay, Serena, Precious, I can see all of you. Okay. Daniel, you're top now. I've seen you. Okay, so while we wait for other persons to join, the purpose of this live class is about your defense coming up on Tuesday or Wednesday. Depending on, I don't know, Tuesday or Wednesday. If there's no change, to be Tuesday or Wednesday. All right, so the purpose of this is to prepare you for that, and I'll be giving you all the tips you need to know. This is totally different from your IT defense. So if you don't take the right step, or please hope you can hear me. If you don't use the right step, you end up flopping. Now, seminar defense, as you know, is a course. I don't know if you can see me clearly. This board is. So I'm waiting for others to join. Just hold on, let me set up something. So this rubber up is not the subject. Okay. All right, thank you, sir. Okay, I believe it's a little, my face is showing a little now. Okay, 25 persons now. I thought by now we'll have up to, say, 100 persons here, but let's continue with the 25 persons. All right, so first, I'll be very fast because I have many slides to attend to already on my WhatsApp now. My WhatsApp is even hanging now because the slides are already more. First, your seminar is a course, 3-unit course. It's a 3-unit course. And what they'll be grading you is your mode of presentation, your slide, the nature of your slide. That was the specific reason why I asked you to send me your slide. So the way your slide looks like, the way you're presenting your slide, that is the way you put yourself as you're presenting, your reports. Those are the things they are going to grade. So before I start looking at your slide, which you've already sent me, if you've not sent me your slide, please send it to my WhatsApp number now. Send your slide to my WhatsApp number if you've not sent it, because I'll be giving you possible questions on your slide and the best way to go about it. All right. For those of you watching with that you've not subscribed, you're just watching, I'll not even attend to your slide. Click on the subscribe button, subscribe first, and there's a thank you button there. If you check up there, there's a star button there. You can drop small thing there first. Let's use and hold ourselves. All right, so that being said, first, your slide. Anthony, is it handsome we are looking at here, or the slide we are talking about? All right, so first, you send me your slide, but defense now. Let me quickly talk about that aspect. Let's assume your slide is okay for presentation. Your slide is better, is good. Now, when it's time for presentation, there's this always this fright that will come up. You're afraid or something. First, you have to remove stage fright. At the end of the video, I'll talk about ways you can use it, proving ways you can use in removing stage fright. So even if there are 20 professors there, you can stand in their front and you defend. Because the truth is, those professors, they have their area of specialization. Your topic they might not even know anything about your topic. So you're there to teach them. Just have that mindset that you're going there to teach them. Because some of them, they don't even know, they don't have idea of the topic you're going to defend to them. Are we following? Some of them, they don't have any idea of the topic you're going to be defending. So when you get there, just have that mindset that these are your students, you're teaching them. How will you treat your students? So just have that feeling. But I'll talk more about this at the end of the video. All right. So the next thing is boldness. I've talked about that a little. Boldness. And some of you, when you're presenting, you speak to yourself or you use bedroom voice. Speak out. Take this scenario. You can explain something to someone. You're actually wrong, but the way you're saying it boldly, they'll be forced to believe you. You can, on the other way, you can be saying something that is right, something that is actually right. But when you the way you're saying it, they'll be doubting what you're saying. And remember, they are grading you immediately. Oluchi, thank you very much. Thank you for your nice comment. All right, they'll be doubting what you're actually saying. Do you understand? So you have to speak boldly, whether you are sure of what you are saying or not. Don't do Be bold. Then when you get there, this is not IT defense. So when you get there, this is how you start. Good day, professors. Good day, my lecturers and my colleagues. Don't say my fellow professors. I say good day, my fellow professors. They first of all minus 20 marks from you. Good day, professors, lecturers, and my colleagues. My name is Excellent Simplified. And I'm here they already know you're in biochemistry, so there's no need saying I'm a student of the University of Port Harcourt, I'm in Department of Biochemistry. You're wasting time. You have about, say, 5 minutes to defend. 5 minutes to defend. Maximum they will give is like 6-7 minutes. 5 minutes to defend, 5 minutes to defend, then they can give you like 2 minutes for questions and answers. Do you get? So you don't have time to be saying irrelevant things. So you're going to just start. Good day, my professors, lecturers, my colleagues. My name is Excellent Simplified, and I'm here to give a seminar presentation on diabetic. This is the first slide I opened. Diabetic retinopathy. Then you go to the next slide. Obviously, which should be your table of contents. Miss Kenan, thank you very much. Essay, I don't know what you're laughing. Okay, so you go to your table of contents. Once you get to your table of contents, some persons, they make that mistake of, whether you call it table of contents or outline of presentation, they are the same thing. So you get to your outline of presentation, and what you do is, don't read out your outline of presentation. Don't say introduction, number 2. Let me check this person's one. Don't say, in course of this presentation, I'll be doing introduction, I'll be doing understanding diabetes, you're wasting time. Your time is going. You might not even finish all your slides before time will be up. So when you get to your table of content or your outline of presentation, just say, these are my outlines of presentation, or this is what I'll be using in course of this presentation. Are you following? Tony, say you're missing me. Are you not seeing me online? You say you're missing me. Must you see me physically? All right, but anyways, we'll see one day. All right, so that's that. Hope you're following. Where should we send our slides to? For those you worked on. Okay, for those persons I worked for, you're not sending your slides anywhere. By the time I start a reviewing slide, you just drop your mat number. Your slides are already saved in my system. So you just drop your mat number and I will check your slide for you. So just hold on. Now, all right, so that being said, after your outline of presentation, you go straight to your introduction and say just go straight to the main points. So why I want to review slide, remove irrelevant things. You don't have time. It's not normal. Maybe it's not a presentation that you're giving enough time. You're not giving enough time. You have just say 5 minutes to defend everything you have to defend and get out. So you don't have enough time. So all you have to do is hit the nail at the head. and move on. All right, that being said, I'm going to start reviewing slide now, and I'll be reviewing randomly. I won't say these are the persons I worked for, these are the persons I didn't work for. Those persons online, I'll review your slides first. Those persons that are not here, anytime they come, they will join us. All right, so, but before I do that, some persons normally ask, if you have any question, just drop it now before I start reviewing slides. If you have any question, drop it before I start reviewing slides. Drop that. While I'm waiting for your questions, let me talk about this. The flyer I talked about, the flyer contains just your table of contents, sorry, cover page, just your cover page. It contains your abstract and it contains your references. That's that about the flyer. Now, take note, abstract is not supposed to be more than 250 words. Your abstract should not be more than 250 words. Now, your abstract also is not supposed to be 2-line spacing. So use 1-line spacing, font size 12 times New Roman. Are you following? Font size 12 times New Roman the font, and 2-line spacing. No, sorry, 1-line spacing. Don't use 2-line spacing for your abstract. And it should not be more than 250 words. Tony, you're asking if there must be a reference. Yes, there must be a reference. On the slide, it's not actually very compulsory, but it's necessary. It's necessary. For all the persons I worked for, I added reference in all your slides. If there's no slide, if there's no reference on your slide and I worked for you, send it back. I will add reference for you. It's not compulsory, but it's necessary. You get? All right. So, and your reference now must be in APA format. 7th edition must be in APA format. I'm emphasizing on it. Okay, Peter, you're asking if abstract is necessary in the slide. Abstract is not necessary in the slide. The flyer will be distributed to all the lecturers present, except there is there's an adjustment this year. But what have been done is the flyers, as you're defending, that flyer will be distributed to all the lecturers on the panel. So as you're defending, they'll be reading your abstract, they'll be looking at your references, if they're in APA format and all this stuff, and they will score you from your flyer also, except there's any adjustment this year. But that's what I've been doing. On your slide, I'm not saying if you put abstract on your slide it's bad. It's not actually bad, but it's not necessary for this particular type of defense you're doing. Do you understand? Lisa said, must there be table of contents on the slide? It's necessary to put outline of presentation. Either you call it table of content or outline of presentation. Yes, it's necessary. And is the flyer a must? Normally, you are not giving orientation this time. My time and last year, it was a must. You print it to as many lecturers are on the panel. That was what was done in my time. You print it to as many lecturers are on the panel. It's not just 3 pages now. So you print it, photocopy to all the lecturers. It was a must then. I don't know how it will be done this time. But for all the persons I worked for, I did your flyer. I did your flyer just in case. Just in case. All right, any other question? Okay, orientation is tomorrow. Good, glory to God. So if orientation is tomorrow, that's a good one. So you hear what they will say from there and you take it off from there. So those persons I worked for, since orientation is tomorrow, if there's any adjustment, don't be afraid, just chat up. I'll effect your correction. All right, so let's move on now. Alice, if you are online, just say hi. Let me see you. If you're not online, I'll move to another person's slide. Because the first slide I open here is yours. If you're online, say hi. If you're not online, I'll move to another person's zone. Okay, while I wait for her, let me open another person's zone. Please just drop hi on my WhatsApp now. Anybody, if you're here and you want me to review, okay, Alice, I've seen you. So let me start from you. Please stop sending hi now. Or yeah, hold on. So it doesn't, hold on, because the chats are going down. So just hold on. Okay, Alice, let me, my battery is low, my laptop battery is low. What do I do now? What do I do? Just give me 1 minute. If you go offline, you miss your turn. Just give me 1 minute. Let me get a charger. Please hold on. Let me charge my laptop so it doesn't go. Let me disconnect everything else. Under the table. There is no one. Okay, okay. I've seen it. Thank you, sir. All right. Sorry for that. I'm back. Okay, so Alice, first of all, from your slide is actually okay. Your slide is actually okay, but I have few comments. I'm seeing your slide for the first time. All of you sending slides, so I'll just look at it and I'll make few comments. Your topic is diabetic retinopathy. First of all, have good idea of diabetes. Type 1 diabetes, the cause. Type 2 diabetes, the cause. Gestational diabetes. Please be jotting down all these things and research on it. So take note of all the types of diabetes, especially type 1 and 2. Learn the cause and anything about diabetes first. Because if Professor Gunka is on the panel that day, that's her area of specialization, diabetes. She will make sure that you answer a lot of questions on diabetes. So have broad idea about diabetes. Okay. Then I feel this your cover page is not well arranged. It's not well arranged to me, but it's not bad. It's not bad to say, but I prefer if you just see what you can do to that. Then your introduction is actually good, but the font size is very small. The font size is small. It's called PowerPoint, not WordPoint. So you're just to have the points, and you're not supposed to have more than one heading per slide. Please, all of you, even if it's not your turn, be taking note of all these things so that you can effect it. You are not supposed to have more than one heading on one slide. So if you are going to the next heading, you move to the next slide. If you are going to the next heading, you move to the next slide. Are you following? Are we following? Say yes or no so that I will know if you are following me. Okay, so all these headings you are putting here, please remove the headings. So you have to touch this slide a little. Then understanding diabetes, you did a nice work here. You talked about type 1 and type 2, but you still made that same mistake of putting different headings on one slide. So adjust that. Next thing here. Okay, you did that throughout your slide. So I assume you have corrected that now. Remove all the subheadings you put. Only the main heading is okay. The other headings, they are not necessary. If you really want to put them, push them to the next slide. Understood? Then work on your font size. Your font size is small. If not, your slide is very good. Your slide is very good. All right, now your references. That's what if Dr. Kwonkwo, the wife, is on the panel that day, her specialty, that seminar defense day is. Those of you still asking where you are sending your slide to, send it to my WhatsApp number. Send it to my WhatsApp. My main WhatsApp number, not the second line, please. So if Dr. CJ Okonkwo is on the panel that day, her specialty should be checking references. Please ensure your references are recent. Use recent references. Stop using 1980 this, 19. If there is any old reference there, maybe it should be like 215. 215 is even old, but you can use it. That's if it's. But use all these 20 something references. Do you get? Like your references here, they are perfect. They are perfect, but they are not in APA format. In APA format, please, all of you take note. In APA format. David Nora, you said I've not sent your slide. I've not sent your slide. Please, after this live class, just send me a message, please. Please. I'll do your own orientation separately if I've not sent your slide. That should be my fault. My apologies. So in APA format, you are to italicize the journal name. So like this is your first any journal name you have there. Like this one now, the first reference there. Diabetes care, that's where you took it from. That's the paper you took it from. Italicize it. The Lancet, italicize it. So italicize all your journal names. italicize all your journal names. Then this reference number 8 here, at least 1991, is too old. Please remove it. You're not born then. Remove it. Then this last slide you put Q&A, question and answer. Now you dey beg for question, oh, pray you answer it. All right, that's that. Next person, please. Next person, please. Next hi. Let me get to my WhatsApp now. Let me get to my WhatsApp now. Just send me hi on WhatsApp. No, on WhatsApp, not here, on WhatsApp, so that it will come up. The first person to come up is the first person I'm checking. Somebody should send me hi on WhatsApp now. The first slide to come up. Okay. Hey. Okay. Special Missy, are you there? I've seen your own. Special Missy, say hi on YouTube. Let me see if you're there. If you're not there, move to the next person. Just saw your slide now. The slide is loading. Where's Special Missy now? I'm not seeing you online. I have 44 persons here live and I'm having just 18 likes. Please, people should stop that thing you're doing. Click on that like button. Click on the like button. The like button. Like the video, please, guys. Like this video. Like this video. Like this video. You're watching for free. This one I'm supposed to do another thing. I'm doing all these ones for free. Special Missy, you send this thing as PDF. Well, let me just do it as PDF. Those persons are thank you. There's also a button there. I don't know if people are not seeing it. Nobody have clicked that button. There's also a button there that you can send money. If you are not seeing that button, have you? Click that button and send small thing. Let me see road. All right, so let's go on. Special Missy, I must commend your slide. It's well organized. It's very, very organized. You did a very nice job here. So my comments on your slide will be few, but your introduction, your work is not justified. Like the introduction is not justified. So just copy it, do Ctrl J so that it will be aligned. Do you understand? So apart from that, please, those persons have reviewed your slide, don't go, because there's something I have at the end of this video. So if you go, you miss that one out, and that might be the most important that will help you. So your topic is integrating targeting in cancer therapy. Know everything about cancer. You know, cancer is uncontrolled growth of cells. So they might even shift you. If Professor Lawrence is there, he might even shift mitosis and meiosis into mitosis and meiosis, because cancer now is uncontrolled growth of cells, cell dividing rapidly without control, and it's mitosis. It's out of mitosis. Mitosis going out of hand. So they might shift you into mitosis. So know all those stages of mitosis: prophase, metaphase, anaphase, telophase, interphase. Know all of them. Know all of them very well, because questions can come from there. Then about integrins, know them very well. So that's that. I will be fast a little so I can attend to everybody. Your references are okay, but they are not arranged alphabetically. Your references are in APA format, good, but they are not arranged alphabetically. Arrange your slides alphabetically. Your references alphabetically. Aside that, you did a nice job here. Next person, nobody should send me hi on WhatsApp. Just hold on, so I won't be biased. Yeah, don't send me hi again. Just hold on until I say send. The next person here. Blessing, if you're online. Blessing of something. Blessing, if you're online, just say hi. Blessing, if you're online, say hi. Not on WhatsApp or on YouTube. Ofue Blessing, something like that. Ofue Blessing, I saw your slide now. I'll move to the next person. I'm not seeing your hi here. Where's this girl now? Okay, so while I wait for her hi, next person, or let me just talk about her slide quickly, because she's there. Maybe she's struggling with network or something. First, your slide. The background is not bad though. It's not bad. The background is not bad, but You know, in that BCH, their projection is normally okay. Anthony, if people are together, I believe she can hear me. The slide is not bad, but in other BCH, like apart from those that will be defending with smart screen, the rest they will be projected. And when they project it, the quality of that projection will be reduced. But your slide is okay. Your topic is biochemical cycling of sulfur in contaminated wetlands. That's a beautiful topic, nice topic with nice questions. Your introduction is okay. Then, okay, just hold on. This diagram you put on slide 6, please know how to explain that diagram, sulfur cycle, because you get a lot of questions from there. So know it. Then importance of sulfur cycling, all those things you put here. Ensure you know it. Ensure you know it. I'm looking for your reference now because that's where. Blessing, you did not put a reference. Please go and put a reference on your slide. Please. Go and add references to your slide. And some of your works are not justified. Like it's not aligned left and right. So justify it, please. Justify it. Aside that, you did a nice work. But if you can change this background, maybe use a plain white background, I think it will be better. But your work is nice. But if you use a plain white background, I think it will be better. Then all this scientific name on slide 11, all the scientific name of Theobacillus, sulfurovum, all those things, italicize them. Any scientific name you're using, please italicize them. All right, that being said, you did a nice work. Some questions you can expect from this your topic: biochemical cycling of sulfur in contaminated wetlands. Know everything about sulfur cycling. That's that. Know everything about sulfur cycling. The next person I'm seeing here, Glorious Prince, are you here? Glorious Prince. Please, those persons I did their slide, I almost forgot. I'm now attending to people I did not even do their work, but I promised to do everybody's own. Please, those persons I did your slide, the way this thing is going now, if I wait until I have to go to your mat number, it will not be easy. So please send me your slide or send me hi on WhatsApp. Send me hi on WhatsApp. Glorious Prince Monachi, your slide is perfect. Yeah, just hold on. Okay. Your topic is hold on. Your topic is monoclonal antibodies and antiviral therapies. Okay, so you have to know what monoclonal antibodies are. You have to know antibody-antigen reaction. Please know that very well. Antigen-antibody reaction is if Professor, what's his name again? This man that teaches forensic biochemistry too. If he's on the panel, you ask him more questions. This is field. So learn about antigen-antibody reaction. Very well. Then your slide is okay. It's okay. I don't have any comment on your slide. It's okay. All you have to do now is master your slide. Then as you're reading your slide, please also read your report, because your report is more detailed. It will give you more idea on what to say. Do you understand? To give me more idea on what to say. Please, if you're watching this, if you're not subscribed to my channel, click on the subscribe button. Let's move it to 8,000 subscribers. I've been on 7,000 subscribers for a long time. Please, let's move it to 8,000 subscribers. If you've not subscribed, do me a favor. Click on the subscribe button. Thank you very much. I appreciate. And let me use this medium to thank all of you that supported me on this my new channel. You know, my old channel, when it was demonetized, I was crying that time. But that channel have been restored last week. But I decided to hold this live class here and not on the old channel. So thank you for your support. I appreciate. All right, so that being said, Glorious Prince, I don't have any comments there. The work is just so nice. I don't have any comments here. Just master your slide. If you have any personal questions, send it to my DM after now, but not this night. I will not reply this night because messages in my DM is scary. I'm not sure I'll even check my DM. Next person, promise. Promise, say hi if you're there. Say hi if you're. I mean, say hi on YouTube if you're there. Promise, please, and please let me clarify this. I'm not being biased here. I'm just picking anyone that comes up from my WhatsApp. So those of you saying hi, please check. I'm actually seeing you, but understand from my own aspect that it will not be easy. But I will attend to everybody. So just stay. Just stay. Just stay on. Okay, promise. This is a promise. This is your slide. Your topic is the role of neutrophils in bone metastatic prostate cancer. Neutrophils, is one type of white blood cell, right? So you have to know the role, first of all, of neutrophils. You have to know what the meaning of bone metastatic prostate cancer. What is the function of prostate in the first place? What's the function of prostate? That's if you should know about that. It's not everything that will be on your slide though. Slide is a guide. You have to have wide knowledge of it. So prostate cancer, know about it. So if you don't know about neutrophils, go and read about neutrophils now. Go and read about neutrophils now. It's on your slide, but read in details. All of you, please go and read in details. Your work don't depend on only what is on your slide. Read anything connected to your topic. Read anything at all that is connected to your topic. Read anything that is connected to your topic. Your slide is actually nice. It's excellent. So I have no comment on that. And promise is not even here. So I think I've said something about your slide. We'll move on to the next person. Next person I'm seeing here is Michael Christabel. Michael Christabel, say hi on YouTube if you are here. Michael Christabel, say hi. All right. So on your slide, what I'm seeing here. Okay, is a promise. I've talked about your slide. Okay, so first things first. Christabel, your work is nice, but your cover page, you have to work on it. You have to work on it. You have to work on your cover page. Is okay, but not well organized. Because the name of the university, seminar presentation, all of them one color, same font. It's not very easy to differentiate them. It's not very easy to differentiate them. This is a promise. It was you now. I've talked about your slide. I just finished. Your slide is the one I just finished talking about. Missy, I don't know if that is how you pronounce it too. So pardon me if I'm not pronouncing it well. Alright, so Michael Christabel, please work on this, your cover page. Then outline of presentation is okay. Outline of presentation is okay. Introduction, your slide is not crowded, so it's okay. Your slide is okay. Just hold on, let me see if there's anything that is not in place. I like the fact that your slide is short, just 9 pages. I'm not saying it must be 9, but some of you that are doing 20 slides, I don't know how you're going to defend 20 slides in say 5 minutes. I don't know how you're going to do that. But if you know you can defend 20 slides in 5 minutes, it's still okay because there's no limit to the number of slides you have to do. All right, Christabel, you don't have references here. Please, it will be better if you add your references. And if you're adding your references, please add it in APA format. And this is your topic, HIV-related lactic acidosis. Know about lactic acidosis very well. Lactic acidosis, how lactate is even formed. They might even shift you into anaerobic respiration because lactate, which is lactic acid, is gotten through that pathway. Pyruvate is supposed to be converted to acetyl-CoA if oxygen is present. But if oxygen is absent now, the pyruvate can follow 2 other pathways. It's either fermented to alcohol, that's ethanol, or reduced to lactate. They get so they might follow that pathway. So have broad idea about that. Promise, you say you don't understand the metabolic pathway. Grace, what do you say I should repeat? Okay. Promise you say you don't understand the molecular pathway. But no, who says he's having problem with? Let me attend to one person first. Okay, lactic acidosis, Grace. So I said lactic acidosis. I said you should read more on it, but they can drive you, they can bend you now to lactic acid pathway. Like this is what I mean. Please, lactic acid is same thing as lactate, just like pyruvic acid is same thing as pyruvate. Do you get? Now, in the glycolytic pathway, all of you are biochemistry students, so what I'm saying now will not be strange. In the glycolytic pathway, glucose follows the 10th step and is broken down. The end product is 2 molecules of pyruvate. Now, this pyruvate now, normally is supposed to be converted to acetyl-CoA through oxidative decarboxylation in the presence of oxygen. So pyruvate is supposed to be converted to acetyl-CoA. The acetyl-CoA then goes into the TCA cycle. But if oxygen is absent, anaerobic respiration will take place. Anaerobic glycolysis, so to say. The pyruvate cannot be converted to acetyl-CoA. The pyruvate can either be fermented to ethanol and CO2, or is reduced to lactate. Which if oxygen or maybe the activities are high, or you're doing strenuous activity, the lactate might accumulate, causing the blood to acidify. That is the lactic acidosis we are talking about. Now, HIV-related lactic acidosis. Your topic now, you should now focus on how HIV can cause lactic acidosis. What in HIV can cause lactic acidosis? All right, so that's that. I don't want to spend too much time on one person's slide because the slides in my DM, there are many, but I'll attend to everybody. And if you go, you won't even know when I will attend to you. All right, so that's that. So this person, add your references and that will be that. So Promise, you said something. You said you don't understand the molecular pathway. So that molecular and cellular pathway, I'm back to your slide, so listen attentively. Neutrophils influence bone metastasis through various ways. One, releasing signals that promote tumor growth, interacting with other cells. And hope I'm reading the right thing from your slide. Okay, yes. Interacting with other cells in the bone environment, modulating immune responses to favor cancer progression. So we are just looking at how neutrophils influence bone metastasis. I believe at this point you already know what bone metastasis is talking about in details, read it in details. So how we neutrophils influence bone metastasis? We said it releases signals like cytokines and enzymes. These enzymes or the cytokines now will promote growth of tumor growth. This neutrophil also interact with other cells in the bone environment and modulate immune responses to favor cancer progression, meaning the neutrophils are in favor of the cancer, meaning remember your topic is the role of neutrophils in bone metastatic prostate cancer. So the neutrophils, they even help in promoting the cancer cells. That's a summary of what is cancer growth, tumor growth. Cancer growth is the same thing as a tumor because cancer is tumor, swelling, the cell division, uncontrolled cell division. I'm sorry, I'm trying to reach everybody. But if I don't spend more time on your slide, just know that you did a nice job. The next high I'm seeing there is amazing. Amazing D. Amazing D. So I've seen your slide. So just hope you're here. Okay, I've seen you. Those of you that you've not subscribed, please, I'm still saying it to subscribe to this channel. If you've not liked this video, like this video. For those of your classmates that are not here, I don't know where they are, but please, only 66 persons are here. I thought by now we'll be having like 200 persons here, but we'll move. All right. Amazing day, your slide as I just opened it. You know, there is a way that, like my own time, I did not even finish my defense when they said I should stop. Your slide will just be appealing to the extent that they'll just be looking at it. They'll be admiring it. Your slide is actually amazing, just like your name. So your slide is okay, but on your topic now, this is what you have to do. Your topic is now generally all of you having all of you that your topic is having advances in something something, advances in something something. Please and please, I'm begging now. They say advances, you are to check from what have been done previously and where we are now. Like in this your own topic, advances in biosensors for environmental monitoring. Advances, when they say advances, like a progression. What has been done, what we are doing now. So read about it. Do you understand? You know, when you're defending, in your defense, you're not to read word for word. You just look at it, you explain, look at it, you explain. You don't read your slide word for word. Are we following? So when they say advances, please, what is the recent thing? Now, where are we now in this biosensor of a thing? So you need to know about biosensors, environmental monitoring. So how is biosensor used in environmental monitoring? You should know about that also. Know about that. So here in your slide, I'm saying fundamentals of biosensors. That is how biosensors work. Recent advances in biosensor technology, very important. So this one now, this is your slide 5. This is your slide 5 where you have recent advances. Please fire it down, understand it deeply, because that is the major thing in your topic. Then applications of biosensors, also look at it deeply, because you'll be asked questions there. Okay, sir. All right, sir. Now, case studies and recent developments, all these things, they are very important. They are very important. Okay. Aside that, if you have specific questions, you can send it to my DM. I'll respond, but not today. So, aside that, everything is in place. Everything is in place. Just work on the recent advances very, very well. Know about environmental monitoring. Most of the lecturers, let me shock you, most of the lecturers in that department, they are environmental biochemists. Professor Belong, Dr. Choma, Dr. Yiri, most of them, they are environmental biochemists. So just prepare for questions here. Prepare for questions here. Lab coachman, NMRO, is specialized there also. So please focus there, because you will be asked questions from there. All right. That being said, the next person's slide I'm seeing here. Peaceman Destiny. Peaceman Destiny. Okay, I'm seeing a comment there. Now, for those of you I did slide for, please. For those persons I did slide for, I said if you open it on your laptop, the view will be different from the way you're opening it on phone. Phone stuff is the resolution on phone, the way it appears on phone might be different from the way it actually is. So if it's scattered on your phone, view it on laptop. I did it with laptop, so it will appear well when it is projected, because it's laptop they will use and project it. So that's that. The chemistry and biology of collagen. Peaceman, are you there? The chemistry and biology of collagen. That's your topic. Sorry, having a headache. I just came from one seminar and rushed down here. That was why I actually shifted it to 8 o'clock, because I was in the other seminar speaking. All right, so the chemistry and biology of collagen. Now, collagen, you have to know detail about your slide. Is okay. I don't have any comment about your slide. It's okay. But what you have to focus on now, know about collagen in details. Why they say the chemistry and biology of collagen is, oh boy, know everything about collagen. They will ask you questions. They will ask you questions. What's the function of collagen? What's the structure of collagen? What is it actually doing? So know how collagen is synthesized. Know about fibroblasts. Fibroblasts. That is where collagens are synthesized. So know about fibroblasts very well. Please, sir, my slide is not ready yet. Please, can I know the possible questions on the topic? All right, just hold on a little, Faith. Just hold on a little. All right, so hold on. Hey, when I on this light is actually affecting my face. Let me see. Yeah, it's better. So that the light will not be affecting my face. All right, so let's move on, please. So, so know everything about collagen. Know everything about collagen. Fibroblasts. Know everything about fibroblasts, and that's that. Just prepare well. Your slide does not have problem. Just prepare well. Know about collagen in details. In details. They say biology and chemistry. The next person I'm seeing there is all of you sending hi plenty times. Just calm down, please. Just calm down. I'll attend to everybody. Just calm down. I dedicated today for you. I was supposed to have another I was supposed to have another class now, so my student is actually waiting for me. He's actually waiting for me as I'm speaking. But I already told him he should wait a little, because this class, until I attend to everybody, I will not stop this class. I will not stop this class until I attend to everybody. I started with you from, I don't know whether you're two, and this is your final year. Even if I'm not around, I will not leave you. I will not leave you. For those persons that have been acting of class, don't worry yourself. Some persons, many persons have been sending messages. I don't even know how this semester is going. I don't. Normally, final year they will not take it serious. I don't know how your own is going, but sometimes 1 week, say maybe like 2 lectures. Lecturers will not even come, and all those stuff. You get? So final year, lecturers will not even take you serious, but exam will take you serious. So that's that. You see, it's exam week. Most lecturers will come and do their thing. That's final year, second semester for you. Even if I'm not around, I'll still do something. So just hold on. Your A in this one. Even if when I'm around or not, I'll do class for you. Your A is settled. I'm done. Amadi Daniel, topoisomerase in treatment of diseases. Your slide is okay. Topoisomerase in treatment of disease. Okay, I did this, and there is a mistake that I was supposed to correct. Please pardon me, I'll do that. Now, this topoisomerase, please know about DNA replication. You know, topoisomerase is one of the enzymes involving DNA replication. So this topoisomerase now that is involving DNA replication, know about it very well. Know about DNA replication. Know about the role of topoisomerase. Then you now come to know about the types of topoisomerase. Then you come to how topoisomerase can be used in treating diseases. Please just understand it. First of all, if you are reading, know what topoisomerase is doing. Mesoma, I'm saying you, please. Gift, I'm saying you. Peculiar, I'm saying you. It's just that once I finish, anyone that comes up is the one I pick. But I will attend to everybody. Now, what was I saying? Know the role of topoisomerase. What is this thing actually doing? Please, to everybody now, please, aside your slide, go and read widely. Don't depend on only your slide. Have broad knowledge. Now, after you've known the role of topoisomerase, now you ask yourself, what is this topoisomerase doing? What is the role of this topoisomerase? What's the role of the topoisomerase in treatment of disease? So you talk about that, and it's actually related to cancer, because since it's involved in DNA replication, topoisomerase, since it's involved in DNA replication, it plays a role in cancer. So please just know that. And this is your slide 7. That will be worked on. All right. Aside that, everything is in place. Everything is okay. All right. The next person, I'm seeing Oluchi there. Oluchi, okay. Oluchi, say hi if you're online. If you're with me, say hi. If I don't see you, I'll move to the next person. If you've not subscribed, those persons that have not subscribed, please subscribe. Oh. And there's a button there you have to press and give me money. I'm not seeing it. Nobody's pressing that button. You are just doing as if you're not seeing it. I'm looking at all of you. By the time this live stream ends and nobody click that button, you will cry. All right. So that being said, Oluchi, your slide, first of all, is good, but I think it can be better. The interface is not very okay. But it's good though, but it's not very okay. I feel you can you can do better. Please, anybody that message me on WhatsApp, please, I will attend to all of you. Just calm down. Just calm down. The messages are just much, but I will attend to everybody, even if it's till 1 a.m. I told you I'm with you today. All right. So first thing, Oluchi, work on your interface, the way your slide appears. The design you use is okay. I'm not against the, but the font you use is not very okay. You can use area black for headings. You can use Times New Roman for normal write-up. Then this is your introduction. You put a box there. I suppose maybe you are supposed to put an image there, but there's no image in that box. Then your work is not justified. So justify it. Justify it. Meaning, if you're using a system to do that, Control J. Aside that, I feel this slide you sent me is not a complete slide because all the space you put for image, I'm not seeing any image there. Then all your headings, use area black or any fonts that can show very well. Then your references, I feel they are old, so work on your reference. Then the journal names, italicize them. Please, all of you, be taking note of all these things. It will be better you correct them before you send me your slide again. All these things I'm saying now, please effect the correction on your own slide, then send it back to me. Do you get? So it will make the work easy, so that I'll be looking at other things. Do you understand? So Oluchi, please work on this, work on that. Then about possible questions, mechanism of lysosomal response to stress. Know about lysosome. Know about lysosome very well. Lysosome is one of the organelles that have lysozyme that breaks down substances that are not needed in the body. So you are expected to know is how those lysozymes in the lysosome respond in time of stress. I just pray they don't shift you to hormones, adrenaline that responds to stress. All right, Stephanie Chiamaka, are you here? If you're online, say hi on YouTube, not on my WhatsApp. This is your slide I'm seeing next. Say hi if you're here. Stephanie, you just sent me that your battery is low, and I saw your slide first. Okay, so if your network is bad, but your work is okay, so your slide is okay, so I'll just give you possible questions and where to concentrate your reading on. Please, I keep on saying, don't depend only on your slide. Everybody, I'm holding my ears now. You can see I'm holding my ears. Don't depend only on your slide. Read broadly. Your slide is just a guide to the defense. Okay, is there? Okay, sir. Your slide is just a guide to the defense. Do you understand? So, biochemical mechanisms of drug addiction and implications for treatment. That's your topic. So, first of all, know everything about drug addiction. Of course, you cannot know everything about it, but know at least what is necessary about drug addiction. Then you now look at where biochemistry comes in drug addiction. Why are people addicted to drugs? You know, some persons, they take drugs, they are now willing to leave drugs, but they cannot. What is involved? What biochemical activity is going on in their brain that makes them addicted to that drug? This will make you talk about neurotransmitters like dopamine. So please read about dopamine. Read about dopamine very well. Read about dopamine. Dopamine is one of the chief hormone that is involved in addiction. Anything addictive behavior, dopamine is involved. Then you look at drugs and some drugs that are some addictive drugs. Do you get? All right. So basically, that's that. So know about drug addiction and the biochemical mechanisms to drug addiction. Then how those things can be treated. How can you just ask yourself, how can I help people that are addicted? How did they become addicted in the first place? And how can I help them come out? As a biochemist, what advice will you give them? That's the implications of treatment. All right, so That being said, you are good to go. Just master this, and with this type of slide, sister, you've gotten A already. If you can present this slide, you've gotten your A. If you can present this slide well. The next time I'm seeing there, Kelechi, are you online? You've sent hi like how many times? Oga, calm down. Okay, Kelechi, if you're online, say hi. If you're online, say hi. People are not responding again. No? If you're online, say hi. Let me see if maybe I'm talking to myself. If you're hearing me, say hi, hi, hi. Or say I'm following, I'm following, I'm following. If you're hearing me, say I'm following. I'm saying 50 persons here, nobody's talking. I'll wait until you talk. If you're following, say I'm following. You know, some persons are struggling with their network. Okay. Okay, let's move on. So, your topic is biochemical insights into the development of biosensors. Biochemical insight into the development of biosensors. So you talk about biosensors very well. You talk about biosensors. You read about biosensors very well. Then for this is your slide. If it is projected on a smart screen, like the one in, if you are defending in this place, where do they call it now? Annex with those smart screen, it will appear well. But if it's projected, this background on your cover page, it will make it not to show well. To make it not to show well, you get. All right, but the other slides are okay. The other slides are okay. So know about biosensors very well. If there are types of biosensors, know about them. Know about them. Your slide is okay. Biochemical insights into the development of biosensors. As a biochemistry, what will be your role in development of biosensors? So before you even know the role you can play as a biochemist, you should know what a biosensor is. Thank God I see this now. References, your references. Don't put, all of you take note, don't put at all in your full references, at all. Like say, just say for example, excellent at all, meaning excellent and others. Put the others in that one, you use it only in text citation. In your full reference, you don't use at all. I keep on shouting this. If Dr. Choma is there on the panel, this is the first thing she will attack you. Please, on your full reference, don't put at all. It is not used in full references. It's only used in in-text citation. So remove all this at all I'm seeing here and put all the names of all the authors. If they are too long, then you can put eclipses. I think that's what they call it. Then put the name of the last author on the list. But I don't think any of them will be as long as that. So that's that. Remove all this at all. Aside that, you're good to go. Next person here. The next person I'm seeing here. Oyeri, Catherine, say hi if you're here, if you're with me. Say hi if you're with me. Please, everybody, just hold on. I'll check your slide. But let me try to be fast a little so that I can attend to everybody. Techniques and relevance to the Human Genome Project. That's your topic. So the Human Genome Project was actually a project. They wanted to sequence all the genes in the human body. All the genes in the human body. And they used 10 years to do that. They used 10 years to sequence the genes in the human body. 10 good years. 10 good years to sequence the genes in the human body. So that's the Human Genome Project. And you know genome. Genome means the totality of your gene. All the gene in your body. All your genetic composition makes up your genome. So the Human Genome Project, they were actually sequencing your genes. And your topic is techniques and relevance of the Human Genome Project. So you talk about the techniques. We have two main techniques. Then you talk about the relevance. You talk about the relevance. How was it important? How is it important? How is it important? So how is it relevant? You know, without that Human Genome Project, most of the diseases that they've discovered cure to today wouldn't have been possible, because like genetic diseases would not be possible at all. So the Human Genome Project was actually a big one. So that's that. Your slide is okay. and I think I'm still owing you something on your slide. We talked about that. I'll send it back. The next person I'm seeing here, messages everywhere. This person, yep. Who is this? Who is this? Okay, this person, Oliver Edit. If you're online, say I'm online. Oliver Edit. If you're online, say I'm online. I'll move to the next person. Oliver Edit. If you're online, say I'm online. Okay, I'll move to the next person. Why? Since she's not here. Anytime you come, we attend to you. Maybe if you're here, say I'm here. Maybe if you're here, say I'm here. If I don't see you, I'll move to the next person also. Is it that people cannot comment again? Hope it's not. If you can hear me, say I can hear you. Let me see if people cannot comment. Because can someone hear me? I just hope I'm not mute. Please, if you cannot comment, please drop. Let me open my WhatsApp. Drop it on my WhatsApp. If you cannot hear me, just drop it on WhatsApp so I can. If you cannot hear me, how will you even hear that one? But if you can hear me, please drop it. So drop it. Let me not be wasting my time. Ruth, I'm seeing your message. Can you hear me? Just drop it on my WhatsApp if you can hear me. I just saw your hi now. Can you hear me? Ruth, can you hear me? Drop it on the WhatsApp. Let me know if you can hear me. Oh, these people. You're just sending hi. How will I know you can hear me? You're sending hi on my WhatsApp. I've called your name and you're hearing me. Okay, so I said you should say hi if you can hear me on YouTube, not on WhatsApp. So I know you're online. Okay, you people can hear me, so why are you not commenting? I'm not feeling lonely. Please, somebody should join. I'm not seeing any live chat here. I don't know if it's on my own end. I'm not seeing any live chat here. Somebody should just drop a message. Let me know if it's my fault. Hey, it's on my own side. I'm sorry for that. I'm not seeing your hi again. It's on my own end. But let's continue. So I'll go back to that person. How do I do this now? How do I rectify this now? I need to be seeing your chat. I need to see your chats. I don't know how to rectify this. I don't know if the chat box is filled, but it cannot be now. Please, let's continue. This person, Oliver Edit. First of all, this is your slide. The size you use. Enable. You have to reformat it. You use that small size. How did they used to call it? You use that small size slide. I don't know how to put it, but use the bigger one, the landscape type. This one is like box. Your slide appears like a square. Use that landscape type, that one that is landscape. There's a way to do that. Go to your if you are doing it on laptop, you can actually adjust that. So that being said, your slide, I feel if you adjust this thing I'm talking about now, it will be better. Okay, thank God I've rectified it. So I can see your messages now. All right, better. So I, just like I said, adjust this stuff here. I think that's the major thing I would say. Adjust it. Then your slide, is not there's no reference. Put your references there. Then I feel it can be well organized. It can be better than this. Justify your work. increase your font size and that. All right, so now your topic is biochemical mechanisms of climate change induced stress in plants. So you'll be looking at how climate change can induce stress in plants and the biochemical mechanism. What is happening in the plant during harsh weather? What is happening in the plant during normal weather? Biochemically. These all your topics are biochemical. So you know it's biochemistry you're talking about. So you have to know that very, very well. And this one you have to read about hormones that can plant hormones that can influence plant development during stress. So read about that. Read about auxin, read about cytokinins, about gibberellins, all those plant hormones, any plant hormone that is involving stress, and how climate change can induce stress. Do you understand? All right, that being said, please, as the class goes on, if you have any specific question on your slide, you can just drop it. Maybe I think I've talked about yours. Have I talked about yours? Maybe. Okay, I've not talked about yours, so let me talk about it now. Maybe first of all, I feel if your topic, you use a different color for this your topic, I feel it will be better. So that, once you project your slide, lecturer should not be struggling to see your topic. It should just appear, it should stand out. So all that is there, they're not necessary. Even your name and math number on this slide, they already have your name and math number there. So it's not from your slide they will get your name and math number from. I'm not saying, sorry. Daniel, you say, we did ask about the work the references did. I don't understand your question. Please rephrase your question. So while I wait for the question, this your work, I mean, maybe now, please use a different color for your topic so it will stand out, so it can show well. Please, if your own is not showing well, make sure your topic stands out. Make sure they can easily see it without struggling. So your topic is biochemical adaptations and challenges of space travel on human physiology. Topic is a strong topic, but you have to do something. Say biochemical is actually an interesting one, though. Biochemical adaptations and challenges of space travel on human physiology. So those persons traveling to space, how do they adapt biochemically, and how is it affecting their human physiology, their body function? Physiology is talking about function of the human body, human physiology. So you have to talk about that. You understand? You heard of the recent story of those persons that were trapped in space, stayed in space for 9 months. Was it 9 months? But I know they were trapped, though they were rescued. What biochemical events will be taking place there? Maybe is your topic, is your own I'm talking about now. So is your slide I'm talking about now. So please, your topic is a nice one. I wish I had something like this, like a topic like this to defend. To be nice. Okay, then the next slide after that is not bad. This is your colorful stuff here, it's not bad. Then you added abstracts. It's not bad, but justify your work. Justify your work. Then the next thing there, I feel this font size you use is small, so please increase it. Please increase your font size. This person is going offline, don't worry. don't worry, I will shock you at the end. You're going offline because I've not gotten to your slide. Please, I still have other things to say apart from this slide review. I'll do this slide review for some time, then I'll pause and give you full gist. Because just like I said, after today, nobody should send me except those I did your work and you need correction. After today, don't send me any question relating to seminar defense. I will not respond. To be very honest, that is why I said today. I have other things to be doing, but I said today. I've spent more than 1 hour here already. and until I finish your slide, this thing will be on. I'm spending my time so that after today, you'll not be sending me slide to review or asking me questions about slide, except it is actually necessary. If it's necessary, I'll respond. But if it's not necessary, I'll not even open it. I'm being honest now. Then maybe another thing you are putting, I'm seeing 2 headings in one slide. This, if you have more than 1 heading on one slide, shift it to the next slide. But your slide is okay. It's okay. Then you did not put references. Please work on your reference. Put references there. Next person. Or this person is saying their battery is low. Your battery is low. Whose battery is low now? How will I know now? Most of your numbers are not saved. So I'm seeing your name on YouTube and on my WhatsApp here, your name is not saved. Next on my own just came up. So let me just talk about your own. Finally, the Lord has remembered you. You should relax. I'm obviously serious headache here. And to click like button is like something is holding your hand. Like this. Like something is holding your hand. And I'm shouting, having a headache. Everybody's battery is now low. Oh yeah, nah. Just hold on. I'll take it fast a little. Relax. Okay. Mess on now. Your slide is okay, so I don't have anything to say about your slide, but let me just give you insight. Calcium signaling in plant biotic interaction. Please learn about calcium. Read about calcium very well. Not like the chemistry of calcium, but what's the importance of calcium to plant? First of all, how does calcium help in signal transduction in plant interaction? Do you get? So calcium signaling. So learn everything you know about calcium signaling. How calcium is. I think I have on my other channel. I made a video on calcium signaling. Please check my other channel. Please, there's some person sent me a message that they were trying to check that my other channel. They were not seeing it. I changed the name. The name is no longer Excellence Simplified. It's now Learn with Excellence. This one you are on now is Excellence Simplified Academy. That other one, that my first channel, is Learn with Excellence. You can go there. I think I made a video on calcium something. I don't know if it's in plant or not, but just check if you see something like that. Okay, so you learn. Your slide is simplified. But read more. So when the plant is in danger, how does calcium transduce signal? How does the plant know? What did you say? So calcium signaling, that's that. I don't have much to say. Master your slide very well. Read your report. Your report will give you details. Then if you have any specific question, feel free to drop it here. Next person, Joyce, say hi if you are there. Joyce, say hi if you're there. All right, so you made the same mistake. Your slide is okay, but you made the same mistake. Don't use this stuff, this box. I don't know how you to call it. This box format. Use landscape. Your slide is this square type. Use landscape. If you are using a system to do that, maybe at the end of this video I'll show you how to convert it. It's not much work, but you have to rearrange, because of if you change it to that landscape, it will. Then this putting course coordinator, Mr. CJ Okonkwo, then supervisor Professor CC Monago. I don't know who asks you people to put the name of your supervisor, your project supervisor in seminar defense. If they told you people to do that, fine and okay. If they did not give you any information like that, please go and remove it. Let me say that again so that you not quote me wrongly. If they did not tell you people to put your project supervisor's name here, remove it. Project is a different course. Seminar is a different course. Your project supervisor is different. Seminar does not have a supervisor. It only has a course coordinator. Seminar does not. For those of you that put project supervisor here, if that is what the course coordinator say people should do, fine, you leave it. But if they don't say anything like that, all those rumors people are speculating because someone actually met me about that, told me about that, and I'm saying it here. If that was not instruction from the course coordinator, don't put yourself in trouble. Go and remove it. But if it's instruction from the course coordinator, good and fine. I don't have anything to say about it. All right. So, though maybe since your orientation is tomorrow, they'll tell you more. So your outline is okay. Just like I said, don't put two headings on one slide. Please, any correction I've made before, even if I see it on your slide, I will not comment on it again because I believe you look at it and then these your references. I've said before, remove all this at all. Please, anything I've said before, that's why you are listening. Don't wait until I get to your slide. Be taking the correction so you effect it. Onyi, calm down now. Calm down, relax. I'll check your slide. It's just your topic here is still joy, rhizo deposits, classification and function. Your topic is straightforward. So you're just looking at classification of rhizo deposits and the function. So it's straightforward. Your questions will not be far from here. Mesoma, you said, what are the key points of calcium signaling plant biotic interactions? I don't understand your question. You mean the point where you have to focus on, or what? Is it where you have to focus on, or what exactly? Let me know. If it's where you have to focus on, I've said you should read everything about calcium signaling. Okay, I said you should read everything about calcium signaling. Just hold on. You say you've been here since, okay? Me, I've been here since too now. Let's understand ourselves. Please, I'm begging. Let's understand ourselves. That's not my fault. So calcium signaling, the molecular component. Just master your slide, read your reports. Your topic is actually a broad one, but God will help you. It's a nice topic, but it's broad. Next person. I think I'll have to take from down now. Let me go down and carry and pick because the way I'm doing it now is just like the most recent people are the people I'm attending to. Why does that have sense? So let me pick randomly from down. Anthony. Ella, just hold on. I know you should just hold on. You've been here since. Don't worry. What I'll be doing now is, I know those persons have been here since. I'll just be picking now. I'll be picking those persons that have been here since, so I can attend to everybody. So Anthony, if you are here, say hi. Eberechi Thompson. Danny Happiness, calm down. I'll check. No, it's not high here. It's on. Just hold on. All of you, just hold on. Can you talk about the font again? The font I talked about, I say font, use Times New Roman is okay. Then for the heading, use area black is okay. Then your font size, there's no specific font size for this particular one, but for your report, use font size 12, 2 line spacing, except the reference and the abstract. The abstract, don't use 1 line spacing. The references, you can use 1.5 line spacing. But your normal work, your main body work, use... No, Daniel Happiness, I'm not picking from... Don't worry, I'll pick you now. Just hold on. I'm picking people that have been here. Since, and not those just coming. Just hold on. I'll pick everybody. Anthony, the rows of ATPs. And you said hi now, then I'm picking from down. That hi you said now, so many highs have topped it, so your name now is even at the middle. So just calm down. The rows of ATPs
a lysosomal function, and autophagy. Your cover page is nice. Your cover page is nice and well organized. The roles of ATPase. Please, this ATPase, though other things are in capital letter, in this ATPase, the ATP should be capital, the ASES should be small letter, because ATPases, this AES it should be small. So that's the first correction there. Please, I forgot to say this one. Please, all your references should be, sorry, it's called PowerPoint. Use bullet points. Don't just write straight. Wow. Use bullet points. Highlight the points. Bullet point. You put 1 point, 1 bullet. Put the point. You put 1 bullet. You put the point. You put 1 bullet. You put the point. So use bullet points, please. Don't just write. That's why it's called PowerPoint, not word point. Use points. You put pom. You put what you're writing. Another pom. You put what you're writing. So Anthony, take note of that. We'll work on this slide. Use bullet points. I'm not saying any bullet points here. Hope you understand what I mean by bullet points. Sir, please can you talk about the font again? I said what for the cover I can use area black, for the other part is slide time. I think I've talked about that now. If it's not clear, let me know. So Tony, your slide is okay. Justify your work. And I feel it's too wordy, like the words are just too much. So reduce your write-up. AI can reduce that write-up for you. Just copy it and ask them to reduce the write-up and still retain the key points. Justify your work and put it in bullet points. Aside that, your work is nice, your work is okay, but there's no reference. Put your reference there. Know about lysosome also. Know about autophagy. Know about it very well. Know about ATPase. ATPase is an enzyme that is involved in ATP generation, hydrolysis of ATP. So know about that. Oluchi, I've reviewed your slide. I don't know why you're sending her again. Don't know why you're sending her again. Adaogo. If I don't pronounce your name well, please pardon me. Adugo or Adaogo. All right. Your slide. Glycogen loading in sports, another malpractice. That's your slide. Glycogen loading in sports, another malpractice. So please, this is your topic. It's very interesting. You have to know about glycogenesis, glycogenolysis, and glycolysis. In extension, know about gluconeogenesis. Glycolysis, breakdown of glucose. Glycogenesis, conversion of excess glucose to be stored as glycogen. Glycogenolysis, breakdown of stored glycogen to release glucose. Gluconeogenesis, production of glucose from non-carbohydrate sources. All those things, they are year 2 courses, and all those things are on my other channel. Go to my other channel, they are in details. They are stored there, they're in details. So you refresh your memory there. Glycogen loading in sports. In sports, some persons they load glycogen so that energy will be stored. So your topic is a straightforward one, and your slide is excellent. It's excellent. So just master your slide. There's not much D here. You already have A already. You already have A. Just present it well, and your A is setting. That's that. Let me move on to people that have work. Your own is settled, like it's settled. Just do it well. Next person, peculiar. Stop crying now. Oh yeah, now let me check your own now, because I just saw your own. Happiness. You're next. Then favor. All right, peculiar. Looking at your slide, it's not bad. It's okay. The cover page is okay. Okay. Your table of contents is okay. Your introduction. Justify your work. pattern and structure. Not bad. Just hold on, I'm trying to point out some things. I like the fact that your work is not jam-packed. Hey, as I say that, I just saw one slide now. This number 7. God. This slide number 7, if you can split it into 2 slides, I think it will be better. Like if you can please split it into 2, it will be better because now they are sorry, I'm trying to do something here. This particular slide is actually jam-packed. This slide 7 is actually jam-packed. So please check that. Please check this slide 7. Other ones are okay. Your references are old. 1977. Oga, were you born in 1977? Were you born by that time? Please, all your references here are old. Your most recent reference here is 2009, which is still old. So please change all your references and put them in APA format. Please do that. Then all this name of the article you are making bold. I don't know if that is the most recent edition, but it's not. Okay, so aside that, that's that. This person, biochemical regulation of methane production in wetlands. I think I've talked about that topic here before. So you're sharing topic with somebody. So if you are listening, I think I said something about that. Oh. Bill Gates, your high is more than 10. Bill Gates, okay, let me check your Bill Gates now. Margaret, what do you say? I don't understand. Okay, Margaret, just I'll get to that. Bill Gates, yours now. Your topic, your slide is amazing. Your slide is appealing. There are some slides I just open and it's just like I will just talk about them forever because it's appealing. If you view all these things on system, you actually know what I'm talking about. If you view it on phone, it might not give you the resolution you want, or it will look scattered. This is looking good. Now, your topic is naked. Since your slide is okay, I'll just give you some hints. Naked DNA, concept and application. So you talk about naked DNA. How you can obtain naked DNA. First of all, you have to know what naked DNA is. Naked DNA, meaning DNA that is not within a membrane. It's not bound within a membrane. That's naked DNA. You know, DNA is supposed to be in the nucleus. And in the nucleus, there's a nuclear membrane in the nucleus. There's a nuclear membrane surrounding the nucleus, sorry. But naked DNA is open. It's just like that. So you have to talk about the concept and the application of naked DNA. So you talk about how to, in the concept, you tell them what the naked DNA is, the methods of obtaining naked DNA, then the applications, there are many here, genetic engineering, gene therapy, and others. So your slide is straight to the point. Read your reports for more information. And if you have any specific question, please let me know. Hey, I'm on the quarrel me like this. So. But the problem here is some of your numbers are not saved on my phone. So even when you are, for those of you saying, hey, or you are first person to send hi now. First person to send hi on WhatsApp now. First person to send hi on WhatsApp. Hey. Okay, this person, let me start the correction from your math number. Your number is not saved, so math number 281. Let me start the correction from the way you saved your slide. Please, all of you save your slide with your math number. And your math number, your math number starts on, for those of you in U20 or U2021 /, but you cannot save with /. So use hyphen. Don't just put everything together. Use hyphen or underscore. Use hyphen or underscore. So this person, Marcus Favor, Chidiebube. Your cover page here, this background you use is okay, but I feel it's interfering with. Big Gate, you say I've not sent your report. Please send me a message after now. Send me a message on my private WhatsApp so I can see it. This other WhatsApp is crowded already. So before I come down and look at it, it will take time. This Favor Marcos, this background is crowding the write-up. Okay, Daniel, Favor, happiness, just sorry, I'll check that now. Aim and objective before introduction. Well, it's not bad, but I don't think it's really necessary. But it's not bad. The rest, yeah. The slide is not bad. Your references, all this at all, please, I feel I've talked about it. Then all these 1970 something references, if you don't want to have problem with Dr. Chioma that day, if you don't want to have problem, please go and remove it. Go and remove it if you don't want to have problem. Saying that being said, that's that. Please, nobody should take it personal this night. I'm not being biased. So I'm just trying to do my best. Margaret, you've sent it how many times? Calm down now. Calm down. If you've not subscribed, please click on the subscribe button, please. I'm pleading. All right, Margaret, it's your turn. Finally, Abi. Your turn is next, Emanuela. All of your turn is next. Just calm down. So starting from your cover page, Margaret, increase all the write-ups there, the font, apart from this main at the top here. This main thing at the top. You now put this format of the cover page. I'm not really okay with it. The format I feel is better. There's no approved or there's no best format, but the one I feel is better. Lisa, you said I didn't say anything about your topic. Have I reviewed your topic? Let me know, please. If I've opened your slide, if I've mentioned your name, let me know. I waste. This topic here, this topic is okay, but I feel the Marcus Favor. Okay, I'll go back to it. Just hold on. So put your university name. You can start with University of Port Harcourt, Department of Biochemistry, seminar presentation. You put your topic, you put your name, max number, you put course coordinator. You get, then increase the font size. Okay, see where you went to put Department of Biochemistry. No, it's not meant to be so. Increase all your font size. They are small. All those lecturers, all those professors that are old, you think they will see all this small write-up. So increase your font size. Then your references. These are not references. Learn about APA method of citation. And this is your last slide. You say questions and discussion. Welcome. When they throw you questions, don't run. Don't run. Now you beg for questions. Me, any slide I'm doing, I don't used to put that questions, please. Don't beg for questions. Before you not put yourself in trouble. You're not asking questions that you cannot answer. Maybe they wanted to say you can go. They now see the next slide, questions, please. They'll now wire you one that you cannot answer. So I don't used to put that one, but no problem. It's not bad. But work on this. But generally, your topic, heavy metal in this protein resource, mechanisms and consequences. Dr. Choma, the husband, Dr. Choma, the wife. This is their area, heavy metals. Their project students, they can actually tell. For years now, all their project students, all they used to work on is heavy metal contamination in primary school playground. That's it. So if they are there, madam, go and read about heavy metals. First of all, know why heavy metals are called heavy metals. Know the different types of heavy metals. Different examples before you even look at their mechanism and consequences on protein misfolding. Then look at the consequences of protein misfolding. What is protein misfolding? the building block of protein, amino acid, that's the primary structure. When they fold, they give us the secondary structure, which can be the alpha helix or beta pleated sheet. Then the secondary structure will interact to form tertiary structure, which is the three-dimensional shape. And the shape, the way they fold will determine their function. And if they misfold, it means their function will be interrupted. With their function, they cannot perform their function again. So you have to read more on that. This person, Marcus, what did you say again? Favor Marcus, I'm trying to. If you are with me, give me that fire emoji. Give me fire emoji. Maybe to cure this headache. Give me that fire emoji. If I see enough fire emoji, I'll be strengthened. So Favor Marcus, I've seen you here again. So your topic, advances in protein folding and misfolding disease. Oh, I just talked about misfolding now. Protein folding, and so while explaining the other person, I've talked about this. Advances in protein folding and misfolding. So I've talked about this. Misfolding, protein misfolding, if it's not properly folded, what disease can it cause? Then advances, what is biochemistry as a department doing in rectifying this misfolding disease or helping patients with misfolding diseases? Advances, I've said before that anytime you hear advances, any topic, if your topic is advances, talk about the recent things. Daniel, happiness, because your fire is actually more. Let me look for your slide. The fire you put there is just like you wanted to burn the house. Daniel, happiness, and I'm not. Send me hi on WhatsApp. I'm not seeing your slide though. The things are just too plenty here. Daniel, happiness, is your turn. Oh, send hi before I choose another person. No. Wait, this guy is. Okay, I've seen it. Like your high is too much. Okay. I'll be fast now. So people are leaving me. Only 31 persons left. No problem. In fact, the lesser the better now. Close to 2 hours already. Okay, happiness. I'm not saying you should leave. Happiness, Daniel. Use a different color for your topic. This thing I'm saying, please, all of you be applying it. I'm not saying everybody should use a different color for topic. If you know your topic is standing out, it's clearly. So as I just look at your slide, the first thing I see is your topic, because that's the most important thing. This University of Port Harcourt, Faculty of Science, Department of, they already know all those things. All they are interested in, there's no time. All they're interested in, they want to see your topic. As you present your slide, the first thing they should see is your topic. So let it stand out. Fructose intolerance and mental health. This topic is a nice topic. Fructose intolerance. Some persons, they have fructose intolerance. Meaning if they take any food that contains fructose, they'll have a reaction to that food. So foods that are rich in sucrose. Now, sucrose is a disaccharide that have glucose and fructose. So, in fact, if you take sucrose, most fructose intolerance patients are also sucrose intolerant, because if sucrose are broken down in the small intestine, the last stage of digestion, if the sucrose are broken down to release glucose and fructose, the fructose there will the person can still react to it. So, this is your topic. You talk about how fructose intolerance can affect mental health. Yet we have many medical biochemists there as lecturers, and many pharmacological biochemists. Professor Bariku, his area, though is not specialized in neurology, but is specialized in pharmacological biochemistry. So expect questions from him. So I like the way what you did here. Fructose intolerance types. We talked about the hereditary is a good one. So master your slides. Your slide is a good one. I don't have any comment on your slide. Your slide is well arranged. Your references, you have a top. Remove all those at all. I'm shouting it now. Please, in case I don't say it on your slide now, miracle, isn't it? Oh yeah, now, yeah. You don't do. I've seen you. Miracle, isn't it? So that's that about that. Please, all this long high will not change anything again. So just calm down. Miracle, isn't it? I'm not saying you on my WhatsApp. Who is miracle using itself? Is it this other miracle I'm saying here? Oh yeah, now let me. I don't know if it's this other miracle, but Odo miracle, send me hi on WhatsApp again. So while I wait for that, and Emali, I've seen you. Just relax. I'll attend to all of you. So just relax. So excellent, I've been here for over an hour. Oh no, actually, the berry. Me have been here for close to 2 hours too, but just relax. I'll check it now. Let me be fast a little. Miracle. This one, Ogechi Miracle. Oh, it's not person's own. Your slide is okay. Nutri Genome, the biochemistry of diet and gene interaction. This person, go and read. This person that have this topic, Nutri Genome. Professor Gunka, let me give you insight now. Professor Gunka, if she's on the panel, you just enter that bunk. This is her area. So you just enter that area now. Go and read Nutri Genome, how nutrition can affect your genetics, how food can affect your genetics. Please go and read read on nutraceuticals. Read widely how nutrients can affect your gene. If possible, check some food substance or nutrients that interact with genetic materials. If possible, do that. Then this is your slide number 7. I feel it's crowded. Your references, they are okay, but you did not italicize the journal name, so italicize all the journal names. That being said, I feel you're good to go. Honor her. Chidi Berry. God has remembered you. Please, all of you, calm down. It's not easy on my side too. But I already promised you I'll attend to everybody, except you leave. If you leave too, I told you I'm having another class after this one. So that class is actually waiting for me. So I'm the one suffering here. So you're just watching and you're not talking. So calm down. Honor Juliet. Rules of non-coding. Effect that correction I said about making your topic stand out. Onyi, you are next. And Odo Miracle is in there. You are next. All of you are next. The roles of non-coding RNAs in diseases. The role of non-coding RNAs in diseases. Okay, so the roles of, please meet me later. Your sister have information for you. So this later. Oh no, can you hear me? Okay, okay. Please, Sarah, hold on. Adishi, I saw your slide. Just hold on. I'll attend to you. Now, this slide now, make it stand out. Shan, Aliyah, you are next now. Calm down, relax. Now, the roles of non-coding RNAs in diseases. First, highlight it and make it stand out. I think I've said that and I've repeated that. All right. So, aside that, non-coding RNAs. You know, RNA, they are single-stranded. They are not double-stranded. If it was DNA, you know, DNA has coding strand and non-coding strand. That's template strand and complementary strand. But RNA, they are single-stranded. So now you are looking at the role of the non-coding RNA in diseases. So if I were you, I'll be very specific. I'll mention specific diseases and how non-coding areas of non-coding RNAs, how they interfere or what their role in those diseases. Then I'm seeing a slide here that have two headings. I've talked about that. Please, your slide should not have more than one heading. So correct that. Your slide should not have more than one heading. So generally, please, everybody do that. If you have old references, please remove it. Remove old references, replace them. You have problem. If you have a on your reference, remove it. You have problem. That being said, I think I've done something there. Odo Miracle is in the. Let me talk about it now. So please, I asked a question. No. Eberechi Thompson, what's your question? Please, let me hear. Then Peculiar said, please, sir, is it the same reference we should? He said, is it the same? He said, is it the same reference we used on the slide that we have to replicate on the flyer? It's supposed to be more. No, it shouldn't be the same. It can be totally different. There's no problem. If it's the same, the references on your flyer should be more than the references on your slide. The slide itself, people I did for maximum there is I put like 5 references or 6 because your slide should not be jam-packed. But on your flyer, you should put like 8, 7, 8, 10, like that is okay. Then in your report, you should have more references in your reports. More references in your reports. Odo Miracle is in your slide is okay. So I'll just talk about your areas you should expect question from or areas you should actually do more on. Mismatch or repair. That's your topic. Mismatch or repair, mechanism, defect, and consequences. Wow, this topic is very nice. If you want to talk about this topic, that is, you actually lecture those people. Emanuela, you are next, please. You're next, please. I've seen your messages a lot. You are next, please. So mismatch repair. You know, DNA has a natural way of repairing itself. I know. Please, you have to know, have good idea about base pairing. Base pairing very well. I have a full video on nucleic acid. Go to my other channel. You know, this channel I'm using now is a new channel. Go to my other channel, Learn with Excellence. I have a full video, a playlist on nucleic acid. All the things you have to know about nucleic acid. Go there, you revise it. That's BCH 305. You did it in year 3. Now, this mismatch repair now, you know, of base pairing. Adenine is supposed to pair with thymine with 2 hydrogen bonds. Guanine is supposed to pair with cytosine with 3 hydrogen bonds. But in some cases, there can be a mistake in that adenine can pair with cytosine, error. And that is that can cause a serious genetic disease. The DNA now, there is a way now of repairing this mismatch, this base mismatch. That's what it's talking about. This base mismatch, now there's a way. So this mismatch repair, you talk about the mechanism, the defects, and the consequences. So your slide is well explained there. Just master your slide. Master your slide very well. And generally now, all of you, as you're presenting, don't read word for word from your slide. You give them that impression that you've not mastered your topic. So you read from your slide, you explain. It's not compulsory that everything you are saying must be on your slide. No. In fact, that's what will make them to know this person really did a nice job. When you're saying things outside your slide, you're using your slide as a guide. Aside that, isn't it? I just saw something now on your slide. And please send me a message. I will effect that. I'm seeing a toy here in your reference. The other authors are there. But please send me a message after now. On my private WhatsApp, not on this my general WhatsApp. This general WhatsApp now. I'm on this after this live class and this after my other class. I'm not opening this WhatsApp today. Till tomorrow. Because it's scary. Messages, I won't even know where to start from. So who is next? Whose battery is low? Precious. Calm down. He said, keep it quiet, don't favor me. Calm down. Animali, you've been here since. I'm not seeing that person that I've been disturbing since. I'm not seeing you now. So maybe your number is not saved. That is why. Onyi, please send me your name on WhatsApp. You've been here since also. I know all of you will yes. On you just send me your name on WhatsApp. Maybe your number is not saved. I don't know. This girl, Enemali Miracle. Your slide is okay. It's good. But I have some things to say. Viral replication mechanism. Your topic is straightforward. So you're just telling them how viruses replicate. How viruses replicate. You know, virus is not a living thing, strictly speaking, because every living thing have a cell, and viruses are cellular. They don't have cell. That is why you've not seen any medication that can cure HIV, because you cannot kill something that is not living. Strictly speaking, virus is not a living thing. There are three things that must be present before you classify something as a cell. First, that thing must have a set of genetic material. Virus have either RNA or DNA, so that one they fit into that one a little. That thing must have a metabolic machinery, that is organelles. Virus does not have any organelle. Virus have only two things: genetic material in protein coat. And the genetic material is either RNA or DNA. Virus cannot have both RNA and DNA at the same time. That's why we have RNA virus and DNA virus. So then the third one, they must have a limiting membrane. Virus does not have a plasma membrane. So in your course of your topic, you talk about the basic structures of virus. You talk about the stages of replication. This thing is well arranged, step by step. How they attach to the host, how they enter, what they do inside the host, how they replicate. You know, you're talking about viral replication. So, and they cannot replicate on their own. Viruses, if you take a virus and put it in a container, it can remain there 1,000 years. Nothing will happen to it. It will just be like that as a non-living thing. But after that 1,000 years, let it come in contact with a living cell, it will start to replicate using the host mechanism, because it does not have what it takes to replicate. But once it gets into a living cell, it will start to replicate using the host mechanism. So what that means, if a HIV patient uses a razor blade, and maybe the razor blade had contact with the body fluid, and the virus is on the razor blade, if the razor blade stays 100 years, 1 million years, that virus is there. If any day another person uses it, that virus, and that person has injury, that virus will get in contact with the body fluid and start to replicate. That is how dangerous it is. So your slide is actually nice, and I have no comments on that. Just master your slide and you're good to go. You're good to go. Chisom, what do you see? Chisom, Onyi, I said you should send me your name on WhatsApp. I'm not seeing your name here. Send me your name on WhatsApp. Just send Onyi. Maybe I save your number differently, or your number is not saved at all. But as I wait for that, Chisom, let me talk about yours. Adishi, I'm seeing you. Just you are close to the next. Hey, I was not seeing this words. Okay, just hold on now. Everybody, hold on. Just hold on. I will touch everybody's on. Close to 2 hours now, and some persons have not yet subscribed. You are just sending me hi. You've not subscribed, as if you are paying for subscription. You're not paying for subscription. It's not MTN subscription. That, madam, stop sending plenty messages now. I've seen your work and I'm trying to open it. Your work is not even opening. Chisom is you I'm talking to. You're still sending more. Your slide is not opening. No. Wait, just hold on. Yeah. What is happening now? Chisom, is it network issue? You said your own will open. It will open. Chisom, just hold on. Your slide is not opening on my system. Let me try another person's one. I don't know why. Let me see if it's a general problem. But I'll attend to you. I will attend to you. Okay, let me try from this person that just saying hi now. Is your lock. The person is on open now, so choose some, just hold on. Or resend your slide. Resend your slide. This person is on now. Gift. Ono, Pasa, Ogene, Yero, all these data people. I beg gift, you know who I'm talking about. Shan Aliyah, what's your name? Why did I save your name? Just send your name on WhatsApp. You've been here since. Let me attend to you. Give say thank God. Thank God. Oh, I like your slide. Oh, I like your slide. It's like old paper. But your name is Boda. Is there in English like Boda? But you understand what I'm saying, Sha? Your name is showing more than the topic. They're not interested in your name because they already have your list printed with your name and your mat number. So by the time they call you to come, your name is already with them, your mat number is already with them. So what is more important here is your topic. So make your topic, increase the font size of your topic, hereditary persistence of fetal hemoglobin. Another person have this topic also, so all of you listen attentively. Fetal hemoglobin. You know, hemoglobin is a vital component of the red blood cell. It's found on the red blood cell, and the major function of the hemoglobin is to transport oxygen. Now you now talk about fetal hemoglobin. Now, we have fetal hemoglobin and we have the main hemoglobin. The fetal hemoglobin is found in children. It's supposed to be the child will grow to a certain age when fetal hemoglobin will not be produced again. Then the normal hemoglobin found in adults will be produced. But in some children, the fetal hemoglobin persists. That's it. Even at that age when they're supposed to produce the main hemoglobin, the system will keep on producing it. Now, so that's what your topic is all about, and it's hereditary. So you have to learn more about that. Learn about the cause. Why is it persisting? How can you help those persons? What can you see? What's the diagnosis? What can you see and you say this patient should have this? If you're a doctor, your references are okay. At least I see few recent references here, so they are okay. Then this thank you for listening. Then you now put some images there. Well, not bad, but it does not mean anything. Okay, Chisom, you've resent it. Then this Chijioke, just hold on. I don't know what is happening here, Chisom. I don't know what is actually happening here. Please confirm if it is opening on your... Emanuela, please send me a message now. I'm not checking directly from my PowerPoint. I'm just checking from WhatsApp here. Chisom, please confirm if your slide is opening. All those persons that... Emanuela, calm down. Calm down. Send a message. Let me send your name. Okay, everybody have been waiting since. So who do I attend to now? Just calm down. Let me make it fast. Who have this topic? This Jane Chijioke. Please, Jane Chijioke. Non-canonical RNA polymerase in plants. So I talked about something like these fonts. Please, all of you calm down now. I know what to do after this slide. I know what to do. Let me start from people that have been here since. After this slide, I'll just be selective now from people that have been here since. Please, just hold on. This is your slide. This person, I'm talking to Ejila Jane Chijioke. This is your... Move to your reference section. Remove all those et al. Make your references more recent. Then questions that can be asked on your topic. This RNA polymerase. Know about RNA polymerase very well. Polymerase enzymes, they are always reading the strands. Read about it very well. Ella will not hear what I'm talking of. Ella, you be talking on the group now. I said send me your name on WhatsApp. I'm not seeing your name here. I'm not seeing any name with Emmanuel like Izaga. I'm not seeing any name like that here. I'm not seeing any name like that here. Send me your name so that I will look at it. While I'm waiting for you to do that, who have been here since? Precious Kobo. Precious, send me your name. All of you are mentioning your name. Please send me your name. Send me your name. Aliyah, Shan. I saved your number as Uchechi now. How do you expect me to know? Aliyah, Shan. Are you here now? Shan, Aliyah. That's what people are using. The name you are using on YouTube, different from the name you are using on your WhatsApp number. I've corrected one thing so many times now. You should go and buy me malt and milk. I'm having a headache. Your topic, I've said before, use this, highlight your topic, let your topic show where. Advanced glycobiology, decoding the human glycome. So you learn about the glycome, learn about it very well. Then I will appreciate if you can, this thing is, this glycome is talking about, is actually complex sugar, glycans, complex sugars. So please read about carbohydrates very well. Then I will appreciate if you can use a white background for your slide. If you can use a white background, it will go a long way. It will be better. Your references are okay. So please ensure you change this background so that it will show well. It will show when projected. If they were using a smart screen, it will be better. But some of the places where some of you will be defending, you will not have smart screen there. It's projector they will use, and that their projector said they are not even projecting on something that is balanced or something. So most of this thing, the beauty of the slide will not be there. Then you put project supervisor there. I've said before that projects have nothing to do with seminar. But if that was the instruction, no wahala. But if it's not, madam, remove it. That being said, I feel your slide is okay. So change the background, justify all your work, do Ctrl J. Justify your work. That being said, that's okay. Next person. The Emanuela, I'm not still seeing you. Abi. Okay. Izaga. Okay. I think I've seen it. Listen. Or is it different? Is it different one? Emanuela. Is it the listen? Listen, is it you, Emanuela? Is it this listen? Talk for. Okay, I've seen it. Izaga. Oh, it's even you, Seth. Ogoni girl that cannot speak Ogoni. If I knew it was you, Seth. Alright, please. Okay, so look at your slide. Your slide is okay. Your slide is good. Chisom Jovia, calm down. Jovita. It's Chisom slide that's giving me issues to open. I don't know why. Emanuela, molecular mechanisms of antibiotic resistance in bacteria. Wow, nice topic. Since your slide is okay, I will just talk about where you are to focus on and possible questions you can expect. Molecular mechanisms of antibiotic resistance in bacteria. There are some diseases that are caused by bacteria. Now you want to kill those bacteria inside you. You use antibiotics. But now the bacteria, they now form odeshi to the antibiotic. How are they resistant to that? Let's take, for example, this malaria. What causes malaria is a living thing, Plasmodium. not the innocent mosquito we accuse. Mosquito is only a carrier, and female Anopheles mosquito is only a carrier, it's only a vector of Plasmodium. Now, when the Plasmodium gets into our system, let's say you are treating malaria, you are giving the drug and you're asked to take. Suzanne, I'm seeing you, just hold on. You are asked to take the drug. The doctor gave you, prescribed the drug for you, but you now, maybe you don't like drugs. You took the first dose, the second dose, then you were relieved. So because you are relieved now, you stop taking. Remember that what the malaria drug is doing, malaria drugs are antibiotics. What it's actually doing is actually trying to kill the bacteria there. The Plasmodium is trying to kill it. So when you take one dose, second dose is reducing the concentration, reducing them, weakening them. Then you stop because they are not strong again. As you stop, those guys will now re-strategize, have more energy. After 1 week, 2 weeks, you fall ill again. You take again, you not complete your dose, you not finish them, they re-strategize. Until all the Plasmodium will now call meeting and say, it's like this person is joking with us. So after 1 week, when we stand up again, you will not bring that same drug. They will not form resistance, they will not form a coating that when that same antibiotic comes, it will not do them anything again. That's why some persons they say, if I take this particular malaria drug, it does not work again. They will ask you to switch. You've misused it. And so the bacteria, the Plasmodium now, is now resistant to that particular drug. So you are to tell them the molecular mechanism of antibiotic resistance. How are antibiotics able to do that? How are they able to form that resistance to the particular antibiotics? Hope you understand what I'm saying. And your slide is well explained here, well explained, no faults, as perfect as 100. So master your slide. Your report have more details, so read more about that. All right, so that's that. Who is the lucky person? The next lucky person? Precious Kobo. Finally. Just pray your slide open, Sha. And Yahichi, not Igbo. So just hold on. I've seen yours. I'll check. But Precious, you sent this slide as PDF. Slide is not supposed to be in your PowerPoint is not supposed to be in PDF format. It's supposed to be in PPT format, PPTX. So if you can convert it to PPT, it will be better, not PDF, so that they will not have issues with it that day. So that being said, also you use that box something instead of landscape. So convert it. First of all, convert it to PPT. Then not PDF. I don't know if you did it like this so it will not scatter. I don't know. But your slide is okay. Your references are old. I'm saying 1998 there. 1998 is old. Remove it. Update your references and italicize. All your references are capitalized. You don't capitalize the references. Please don't capitalize it. Work on that. Aside that, your slide is okay. Plant mitochondrial respiration, energy source and energy use. Wow. Just pray. Professor Sien should be on the panel to give you more questions because these are area. Professor Sien, plant mitochondrial respiration, energy source and energy house. You know, mitochondria is the powerhouse of the cell where energy is generated. So you have to know about cellular respiration, how cellular respiration take place. And that might even lead you. They can even ask you questions about the TCA cycle, tricarboxylic acid cycle, also known as Krebs cycle, because it takes place in the mitochondria, and it's one of the ways in which energy is generated in the mitochondria. So please work on what I've said. Aside that, your slide, it's not bad. Next person. Who is the next person? I'll just take, oh God, what will I do now? Suzanne, why are you crying? Don't cry. If there's anybody to cry, here is me. because I just finished one seminar before starting this one. Having a deck already. This one is over 2 hours, and I'm having another one after this one. So don't cry. It's me that's supposed to cry now. And people are not even clicking that button. All right. Suzanne, your slide is okay. The slide is okay. Glial neuron interactions in frontotemporal dementia. Okay, I saw your message earlier. Before this live stream. So I'll work on that and send back. But it does not change anything. So I'll just work on that and send back. Right? So this one now is actually neurobiology. Is about the brain. So you have this frontotemporal dementia. Is a brain disorder that affects behavior. The person will just be behaving somehow abnormal. Who will be on the panel that? No lecturer there specializes on neurobiology, neurobiochemistry. Though we have medical biochemists there and pharmacological biochemists. So Professor Patrick, Professor Barico, all of them, they can have what to say here. So please, this topic, prepare as if you want to teach them, because most of them that will be on the panel, they're environmental biologists, biochemists, and they don't know much about this topic. So you're on a better stand. I'm not saying you should relax, because all those medical and pharmacological biochemists, they can wire you questions here like this, and before you know yourself. So, master, know what dementia is. Then know what neurons are. Neurons are used in transmission of information in the nervous system. So neurons are the functional units of the nervous system. So you talk about that. Then you talk about this part of the brain, this frontotemporal region of the brain. So you talk about it. Talk about it where. Then talk about glia and neuron. What is the relationship between glia and neuron? Glia, they interconnect the neurons, sending information to the neurons. Neurons, they help in transmission. Look at neurons as electric cable, transmitting electrical impulse. Just look at it that way. I started that. Your slide is okay. Next person. Who is here? You saying your battery is almost dead. You've been here since though. And here something. Alua is your turn. Please, everybody calm down. I will attend to you. Alua is your turn. If you're not subscribed, click on the subscribe button. Alua, no now. I've talked about this topic now, so you should have taken mismatch repair and cancer. I've talked about mismatch repair. Maybe you're not paying attention because it was not your turn. So you're actually sharing this topic with somebody. I've talked about mismatch repair. Jurua, favor, please send me a message on WhatsApp now. Mary May, I'll check your message now. So mismatch repair, your slide is okay. Your slide is very perfect. Then I've explained something about mismatch repair before. How DNA is double-stranded, how base pairing. I talked about it not long ago. So you can maybe shift. You can go back a little to check when I talk about that, so I can attend to others. But I talked about that not long ago, a few minutes ago. Another person's slide. Next person. Who is here? Who is the next? Favor, I'm not seeing your message here. Favor, I'm not seeing your message. Just send me hi. Chisom, you sent 52 messages, only you. Just hold on, I'll check it now. It's just that your slide was not opening. Chisom, you are still sending. Only you 52 messages. Calm down now, calm down. That will not change. Oh yeah, stop crying. The defense is not tomorrow morning, Abi. Or what do you suggest? Should I hold another live class tomorrow? Just drop it in the comment section. Should I hold another live class tomorrow? I didn't plan to. Should I hold another live class tomorrow? Let me see it in the comment section. I'm waiting now. Should I hold another live class tomorrow? While I wait for responses from majority. Chisom Judith. First, I've corrected this before. You use this four corner something. Make it landscape. Make it landscape. Then your topic is endorphin release and biochemical basis of the three in Bongi jumping. Okay. People are just confusing me. I don't even know what to do. But calm down. I don't plan holding. I'm having another thing to do tomorrow by this time. I'm having a class by this time tomorrow, so I cannot hold it tomorrow. Let's finish it today, please. Let me know I've sacrificed today for you. So, your topic, Odo Miracle, I'm coming back to you. Endorphins. You talk more about endorphins, how they are released. Endorphins are hormones. So you talk more about it, like talk about it in details. Talk about it in details. Talk about it in details. When I say in details. I follow you. Talk about it in details. Hold on. Your slide 6 is a little jam-packed. Please justify all these things. Justify them. Then your references. I'm seeing a thought there. Remove them. And that's not the format of. Okay, though is the last two references are not in place. So it's that you change them or you work on them. It's that you change them or you work on them. Aside that, your topic is a little somehow, but master your slide and do more research in your report. You'll be good to go. So change this four corner stuff to change it to landscape. If you're still here with me, Chisom, the problem I'm having is your slide is not opening on my system. Your slide is not opening on my system. So Chisom, was that the one that did your work? Just drop it in my WhatsApp. Not on YouTube. Just on WhatsApp. So let me see if I can retrieve it here. Your mat number. Okay. Just hold on. Let me go to my PowerPoint. Your mat number is 30. Let me see if I can check it directly. Just hold on. So we are just talking about the fact that you've been here since. What of me now? This is a free class. I hope you're aware. I'm not paid for this. It's a free class. So you're supposed to pity me more. So calm down, please. Calm down. 5535030. Okay, Chisom, are you with me? So finally, I've opened your slide. ATP is in plant cell homeostasis, the role of proton pumps in nutrient uptake. ATPs, I've talked about ATPs. So you have to know about this enzyme very well. ATP is the energy currency of the cell. Just like Naira is the currency of Nigeria, ATP is what the cell recognizes. So ATPs is an enzyme. In biochemistry, any word ending with ASE, just have that hint that it should be an enzyme. So ATPs is an enzyme involved in the catalysis of ATP production. Now, plant homeostasis. Homeostasis, maintenance of stable internal environment. That's homeostasis. Now you are looking at the role of proton pumps in nutrient uptake. So proton pumps in nutrient uptake. What are the roles of proton pumps in taking nutrients, uptaking nutrients? So you learn more about the roles of ATPs, proton pumps in plants. Your slide is well arranged and well simplified. So take your time on it and read your reports very well. Don't say you're not going to defend your report and you neglect your report. Read your report in details. ATPs in plant cell homeostasis. They won't ask you much question from this. So you have a better stand already. But go and read your reports very well. If you have a personal question, okay, I think let me fix tomorrow let me not. I wanted to fix another time tomorrow where we'll just respond to questions on WhatsApp, but I have other things to attend to, so I will not be able to do that. Who is Didi? Didi is your turn now. Chisom Jovita, I think I've responded to that. Was it Chisom Jovita? I just finished it now. Away, this person, you have work to do. Didi, you use Microsoft Word, then you converted it to PDF. You have work to do here, please. This is not PowerPoint. I saved your number as Didi. I don't know. The name I'm saying here is Chidima Amarachi. Ugoike Chidima Amarachi. I don't know if you're online. Advances in breast cancer chemotherapy. You have to use PowerPoint, not Word, like not Word document, PowerPoint. So you have to redo this thing. Yeah, thank God you joined this live stream and I'm saying this. Just thank God. They won't even listen to you on the panel because it's not PowerPoint. This is Word document. So that's that. Go and attend to that. Then read about cancer very well. Chemotherapy, what can you use in attending to breast cancer? Then advances, what is the recent thing they are doing? What are the recent things they are doing now to attend to that? All right. Your references are okay, so work on that. Next person. Mary May. Somtochukwu Ijoma, please send me a message on WhatsApp. Somtochukwu. Obinwa Kenneth, I'm seeing you, please. And Somtochukwu Nubia, I'm with you also. Thank God people have reduced. Remaining 16 persons, thank God. So send me a message on WhatsApp. Okay, thank God, I'm back. Chidima Marachi, I've talked about your stuff. Favor Mauzo, send me a message on WhatsApp. This person now, Mary May, recent advances in glycomics, unraveling the glycocode. Your slide is okay and well arranged, very consistent. Then this glycomics, read about glycocode. Anytime you hear glyco, first thing that should come to your mind is sugar. Glycans are polysaccharides. Are you hearing? Anytime you hear glyco is talking about carbohydrates, complex carbohydrates precisely, like glycogen and the rest. So this glycocode and glycomics is talking about is just revolving around. And glycomics is the study of sugars attached to proteins and lipids. So any sugar attached to proteins, if you are studying sugar attached to proteins or lipids, what you're doing is glycomics. So glycomics is actually a study of sugars attached to. So your topic now, you are looking at recent advances in the study of sugars attached to proteins or lipids. Because any sugar attached to protein or lipid, they're actually doing something there. They're actually doing something there. Do you understand? So your topic is where your slide is well organized. My advice is read the report very well, because what you see on the report, you will not even see it here, and that will give you insight into more. I believe with that, I've been able to say something. I'm sorry, I'm actually rushing so I can touch everybody's zone. This person that is sending hi now, who is this? Okay, Ijoma. Ijoma, Ijoma, you're next, please. I've opened. Okay, Jessica, just hold on. Chisom, you that you are doing voice recording now. I cannot, I cannot listen to voice recording now. I cannot listen to voice recording now. This person, Oluwari Ibiapuye Tom, something like that. I just opened that one, so I'll go back to others. Pharmacological chaperones as a therapeutic strategy for cystic fibrosis. This chaperone, read it well, because that is Professor Bariko. His area revolves around that chaperone of a team. So, and this your cover page is jam-packed. Your cover page is jam-packed. Your table of contents. Brother, your slide is 25. Is this brother or sister? 25 slides in 5 minutes? I'd love to see the magic. 25 slides. I feel your slide is too much. It's too plenty. Those persons I did for the highest I did is 13. If not, majority 11. So I will say, if you can remove some slides, things are not really necessary, remove them. Just leave the important points. So this person, please work on your slide again. Remove things that are not necessary. If your slide is now more highest, it should be 15. That is if it's too plenty. So one is 25. So find a way to remove some slides here, please. Just my suggestion, though. It's my suggestion. So that being said, Ijoma, Chisom, I've responded now. Chisom, are you not the one I attended to? No. Are you not the one I just attended to? You sent more than 100 messages here. Have I not attended to you, Chisom? How many Chisom do I even have here, sir? Mark number 30, I've attended to you now. So which message are you still saying now? Please make your point clear. Make your point clear. I've attended to you. Maybe you're not really live. Like, click on the live so that maybe you see that I've attended to it. Click on live so that you get to where we are now. Ijoma. You said they can. Vida, I'm seeing you. Kenneth. Okay, Kenneth. Ijoma, God's favor, I'm seeing you. Please, you guys should just help me. Give me a little time. It's not easy on my side. It's getting to 3 hours. Thank you, Santochukwu. Ijoma Daniel Santochukwu, I love this your topic. On a mission to save debt, Cascade. I like this your topic. On a mission to save debt, Cascade is a nice topic, and your slide is well articulated. Now, this programmed cell debt, there are some cells that have been programmed that so time they will die. It's called programmed cell debt. And programmed cell debt, the term used for programmed cell debt is called apoptosis. That's the first thing they can ask you. That this mission to save debt, what is the scientific term for it? This programmed cell debt is apoptosis. It's written on your slide. Apoptosis. Now, These the steps, the proteins involved in apoptosis in programmed cell death is they are called caspases. So you are looking at the series of reaction that take place in programmed cell death. So read more about apoptosis and read more about this series of steps involved in apoptosis. You have a nice slide here. You have a nice slide here. So read more about apoptosis. Read more about apoptosis. Work on your slide. Work on your read your report. That's what I wanted to say. Read your reports very well. That will be that. Kenneth Obinwa. The first thing, rename your slide. Rename your slide. Use your math number to save your slide. That is the format you are going to be sending it. So rename your slide as your math number. Understood? I believe it's understood. The role of AI in protein structure prediction, AlphaFold insights. This is where bioinformatics comes in. Bioinformatics. So the role of AI in protein structure prediction. This is actually a recent area that is advancing. So you talk about recent advances. Please justify this work, Control J, and don't make it too crowded. Justify the work. The content is okay. Content is okay. Your slide is okay. Your references, I'm seeing a thought there in one of the references. Remove it. Remove it. Aside that, just know the rule, what AI is doing in how to predict protein structure. Just know the role of AI. Your topic is straightforward and a good one to defend. Ejerua favor. Are you online? Ejerua favor. Your slide, you use a box, you use that, convert it to that landscape. Convert it to landscape, like that's a bigger one. Convert it to that bigger one, please. Bacteria toxigenic diarrhea and electrolyte replacement therapy. Learn about diarrhea. Learn the toxic gene that bacteria, the toxic role of bacteria in diarrhea. And learn how the electrolytes you can use in the replacement therapy. So you learn that. Master your slide. I don't know how your report is, but make your reports well. Then this slide 4, this is your slide 4. There's how it is appearing here. Check on your system. I don't know if that is how you made it, but if that is how you made this your slide 4, you have to work on it. Something is appearing outside the slide. Then just master your slide. Some of them you have to increase the font size of some of them. Don't have 2 headings on one slide. I've mentioned that before. Don't have more than 1 heading on one slide. Then your references. Italicize the journal name. And I feel that is okay. That's okay. Next person. Jessica Nubia. Liposomes as carriers for drugs and enzymes. Some of you, once I review your slide, you just exit pap. On you, I've not still attended to you. On you, send me message now. Some of you, once I just attend to your slide, pap. Thank you, you not see. You just exit prap. Oh wahala. So Nubia, your turn now. Effect this correction I said. Make your topic show very well. Like make your topic show very well. Make your topic show very well. Favor ones have I not attended to you? Make your topic show very well. Then this outline of presentation is okay. Introduction is okay. Your references, okay, they are okay, but italicize the journal names. Italicize the journal names. Possible questions they can ask you from here, or area you have to concentrate more on. Liposomes as a carrier for drugs and enzymes. Now, liposomes, how do they carry drugs? How do they carry enzymes? So your topic is straightforward. So learn more about that. Read your reports in details. If you have specific questions, feel free, let me know. If you have specific questions, let me know. Who is this person? Tom, I've attended to you now. Who is this person? Favor, wow, okay, it's now I'm seeing your work. Ejerua, favor, I've attended to you now. Ejerua, favor, I've attended to you now. I've attended to you. But this other favor, Monzo. The same thing. I like the flower in your slide. It's beautiful. But make it landscape. Blessed. Enyong, you've not sent your slide and you're saying send your slide now. How do I review it if you don't send? Favor, Monzo. Make your slide this landscape, not this box type. I don't know if you understand what I'm saying. Drug delivery system, liposome and nano carriers. This topic is similar to the last slide I reviewed. It's a straightforward topic. Drug delivery system. How is drug delivered? What is the role of liposome? What is the role of nano carriers? Nano carriers, they are small molecules that help in carrying those drugs. Overview of drug delivery system. Not bad. It's not bad. This slide 5 I feel is jam-packed. So basically what you have to do is reorganize your slide, like make it that landscape of a thing. Then your reference, work on them. I'm seeing et al, and I'm seeing some old reference. Your most recent reference here is 2017. Please work on it. Chisom said, what did I say again? Hey, Chisom. You have to hold on now, Chisom. I cannot resend it now. Just hold on after the. But I've said something about your slide now. I've said something about your topic. But just send me a message after now. I will resend it to you. It will open. Just hold on, it will open. Ihetegu Covenant Sontochukwu. I'm with you now. Work on your cover page. Your cover page is not in order. Work on your cover page. The outline of presentation is not bad. Then introduction, work on it. I'm not saying it's bad though, but increase your font size. Mary May, I've talked about your work now. Mary May, I've reviewed your slide. This person, I'm reviewing your slide now. The major thing you have to do here is to organize your slide very well. That's the major thing you have to do now. Organize your slide. Put it in order. That's the major thing you have to do here. Put it in order. Your font size and the rest. Put it in order. Put it in order. That's the major thing you have to do here. Your topic is straightforward. Your topic is straightforward, but organize your slide well. That's that. This person. Akani Destiny Chika. Cellular senescence in aging and diseases. Senescence is talking about aging. I like the fact that you use a white background, but your slide is not really well organized. Increase your font size, Make your slide, just rearrange your slide, make it well organized. I'm suspecting you use a phone to do this. Please rearrange your slide. Rearrange your slide. Your references also, they're a little old. Use some recent references. And you are not to number your slides, slide 1, slide 2, slide 3. It's already automatically numbered as you're going. So there's no need numbering slide 1, slide 2, and the rest. Your topic is a straightforward one. They might ask you the role of antioxidants and free radicals. They might ask you the role of free radicals in aging. So read about free radicals, read about free radicals, read about that. Jessica. Covenant. How many persons are you sending now? I thought I've opened your slide. Santochuku, Nubi, I've checked your own now. Okay, Ologe, you said it's a tour permitted in your flyer. If so long as it is full reference, the full reference section, a tour is not permitted. So long as the full reference, nowhere, even in projects, nowhere, a tour is not permitted anywhere. A tour is not permitted anywhere except it's in in-text citation. In-text citation, no wahala. But so long as it's full reference, it's not permitted. Vida, have I not checked your work? Who is this person? Delphine, aquaporins and water stress adaptations in plants. Your topic is straightforward now. Just know about aquaporins, what they are doing, the role of aquaporins. Aquaporin is actually protein, so know about the protein and what it is doing in response to plant stress, water adaptations in plants. So your topic is straightforward and your work is organized. All you have to do is master your slide and read your report in details to have more information there. Who have I not attended to? Okay, Vida, let me check your slide after this one. Wisdom, that is blessed, Enyon. Naked DNA, I've talked about this one, so I'm not talking about it again. Somebody shared this topic with you, and I've talked about it. I talked about it in details. Naked DNA concept and application. But your slide is okay. Just make the topic to show where. Make your topic to show where. Italicize the journal name in your references. Then update your reference, make it more recent. But about the topic, I've talked about it before. Someone shared that topic with you. Vida, it's your turn. No, Mary May, I've checked your work. Maybe you were not online that time. I've reviewed your slide for you, Mary May. I've reviewed your slide. Hey, Jesus. Vida Eberechi, extracerebral metabolism in comatose patients. Someone actually defended this topic last year, and even this year, someone is sharing this topic with you because I remember I worked on this topic. Extracerebral metabolism in comatose patients. So you know about comatose patients, then you know what coma is and what can cause coma. So then extracerebral metabolism outside the brain, outside the cerebrum. No, cerebrum is the seat of consciousness. So if someone is not conscious, it means the cerebrum there's a problem. Please, Chisom, make what you're saying clear now. You say I don't I'm talking to many persons here, so I don't know what I've said about your topic. What did I say that you did not understand? Please let me know what I said that you did not understand so that I can recall what I've said already, because I don't know. I can't remember what I said about your slide. So please let me know what I said that you don't understand or what you... But I've reviewed your slide now. About the sending, I said I will resend it. So this person here, work on your references, update your references, make it recent, remove a all. Aside that, your topic is straightforward and it's okay. Your slide is okay, just work on it. Your table of contents here, well, it's okay. It's okay, just correct the few things I've said. Then make your topic maybe a different color or something that can highlight it, that can make it show well. Cardilobary. Onyi, I don't know if you send me a message. I don't know. I can't know. But send me a message again. Let it come up. Because you've been here now. And other persons have been here too. So Cardilobary is your turn. Please check. Okay. Your slide is making sense. Lead toxicity and its molecular pathways in humans and ecosystem. Your slide makes sense. But I just hope they see the topic clearly. Contents, your table of contents is okay. Introduction is okay, but just that I feel the write-up is small. The next slide is okay. So your topic is actually very straightforward. And you also did a nice work. Then in your reference page, all those places all those at the end of the slide, I'm saying full text abstracts, full text. Please clear those things, remove them. Clear them, remove them. They are not necessary. Your slide is okay. Your slide is okay. I don't have much to say about that. Next person. Peculiar. Essay on ye. I'm not seeing your slide here. Peculiar Marachi. This design you use, personally, me, I don't really like it. Your work is okay, but this, especially this cover page, is blurring some part of the things you wrote there. So if you can change the design, it will be better. Fit 2, I did not block you. I saw it. I will check it out now. How will I block you now? I hold you. Don't bold the references here. Don't bold it. So if you can change the design, it's okay. Then RNA vaccine, someone shared that topic with you and I talked about it. So okay, Ruth, you are the only here. Uh, oh yeah, Ruth, let's talk about it now. Chisom, what do you say? Chisom, have I not attended to you? Only advances in plant lipid biochemistry. Go and read about lipids very well. You know, lipid is a course on its own, and Dr. The course coordinator self is Dr. CJ Okonkwo, is the owner of that stuff. He knows lipid in detail, so go and read about lipids. Implications for biofuel production, as is used in biofuel production. You have a nice slide, a powerful slide for that matter. So I won't have much to say about it, but go and read about the advances in plant lipid biochemistry. Advances in plant lipid biochemistry. Know about lipids. They can leave your topic. Most of those people, they used to be funny. They can leave your topic and be asking you details about lipids. About lipids. So at least have good knowledge on lipids. Then know about biofuel. Aside what is in your slide, read the step by step on how biofuel can be produced from lipid. How biofuel can be produced from lipid. So that's that. You have a nice slide. Who is still here? Who is still online? Who is still online? Who is still online? Faithful, I'm looking for your slide now. Please, if you're online, see, let me know so that. Who is this person? I don't know if this person have gone on offline. Nobi, I've talked about your topic now. Mary May. Oh, let me go back to it now, but I've talked about it. Effect of environmental pollutants on human epigenetics. The person that have this topic now, your number is not saved on my phone, but just listen to your topic. Effects of environmental pollutants on human epigenetics. You did not put cover page there. That is the only thing on your cover page. No name, no name, no math number, no course coordinator, nothing. So please work on that first things first. Then effect other corrections I've said on from other person's work. From other person's work. Uchechi, Basilia. Biomarkers of environmental pollution. Recent advances in detection. Your slide is okay. Then you said something about the diagram. You people have crowded my WhatsApp. My WhatsApp has started hanging. My WhatsApp is hanging at the moment. Okay. So this diagram on slide 4 here is just pollution biomarkers can be used to assess the nature and extent of pollution. It's just as explained as it is. Can be just as it is. And your slide is okay. Who is the next person? Who is still here? Let me not be talking about people that are not here again. Mary May, you're giving me 2 times work because I already talked about your work. But let me talk about it now. So Mary May, look at it now. Mary May, are you still here? Okay. Just hold on, I'm trying to open it again. Peculiar. You're giving me 2 times work. Let me talk about people I've not talked about. It's almost 3 hours and I'll be ending this class soon. Oh, Mary May, I spent time on this your topic. It's now I'm even seeing it. I spent time on this your topic. Chisom, what do you want me to do for you? Please, what do you want me to do? Please, please. Just make your point clear. Have I not reviewed your slide or what? Let me know, please. Anthony is the second one that is correct. Is the second one that is correct. Recent advances in glycomics and unraveling the glycocode. I said glycomics is actually the study of sugars that are attached to proteins or lipids. So glycomics, you are studying the sugars attached to, then glycocode, just your slide is already well organized. So read more about glycans, read more about carbohydrates, read more about carbohydrates. I think that's the first time I spent time to say something about it. But now I'm tired already, sir. It's 3 hours now already. I'm talking for 3 hours. So Tochuku, the next one you sent. Tell anybody that have that slide. Azuka, Francis. To rearrange the slide. Make it this landscape type I talked about. Tell the person to rearrange it. Then the topic is not showing well. Make the topic to show well. This introduction, the write-up should be clear. Then this font you use, this Nigerian something font you use, let the person use a different font. Times New Roman is better. Then okay, all this write-up, then the picture taking the major the space, then write-up staying at the side is not very good. So the person should work on that. Then the reference et al, remove all those et al. Then update your reference to put more recent references. Chisom, you are not answering me. You are just doing your own. What do you want me to do? Have I not attended to your slide? If I've not attended to your slide, send it to me on WhatsApp. It's not to be saying I'm waiting. There are many persons I'm attending to here now. So if I've not attended to your slide, send me on WhatsApp. Just send me a message on WhatsApp. Tell me what you want me to do. Because like this, I'm confused. Faithful. Anthony, yes. The size of the case of the letter, that's it. Faithful, your slide is not in PPT format, it's in PDF format. Please, if you can change it to PPT, that's the mode of presentation, not PDF. It's PPT or PowerPoint. And this course coordinator is not Gunka. The course coordinator is Dr. CJ Okonkwo, Mr. CJ Okonkwo. That's the course coordinator. What size should I use on the write-up? The size you can use does not have any size, any preferred size, but so long as it's big for people to see. So, and faithful, you have 2 cover pages. Please reduce it to 1. Then change it from PDF to PPT. Phosphate pollution, biochemical and ecological impact. So you just look at biochemical impact of phosphate pollution, ecological impact of it. So your topic is straightforward. Your references are too old, 1999. To work on your references, remove a top. That's that. That being said, next person. Next person. This person, Oyeri Catherine, relax now. I will do it. I will send it. I've done it several. I will send it to you. Cannot be doing two things on this slide now. This live stream is coming to an end. If I've not attended to you, please drop it on my WhatsApp now, because I'm ending this live stream soon, like soon. If I've not attended to you, drop it now. Before a year, I was on the live stream. I did not review your slide. I was on the live stream. I did not review your slide. Miracle. This other miracle. Have I reviewed this slide? Have I reviewed this slide? Please, those of you still online, if I've not reviewed your slide, please. If I've not reviewed your slide, let me know. Esther. This Esther. On you? Okay, on you said I've talked about your slide before now. Don't worry, I'll go back to it. Esther. Let your slide load. God, I'm tired. Okay, if there's another Chisom here, please, you Chisom, this other Chisom now, send your work again. Just send me a message on WhatsApp so that it will come up. Esther, the role of tight junction proteins in maintaining blood-brain barrier integrity. Blood-brain barrier integrity. I think there was a problem here to add B to this stuff. I don't know if I've sent it again. If I've not, let me know. So, blood-brain barrier. Learn about tight junction proteins. Your slide is okay. Learn about tight junction proteins. Learn about blood-brain barrier. Very well. Nobody specialized in neurobiology there, so they won't ask you too much question. Your own is just to defend your work very well. Then this is your slide 6. That diagram there. Those are parts of the protein. Those are parts of it, of the tight junction. That's how tight junction looks like. So you should be able to use the diagram to then just look at the diagram. Be bold. In this diagram, you can see that is catenins, that is ZO proteins, that is occludins. If they ask you what's the function of this one, tell them sir. With boldness, so tell them sir, they are all proteins involved in fact their functions are ESF. Their functions are on your slide 5. So I think a nice work was done here. So just master your slide. Everything is, you're good to go. Master your slide and you're good to go. Roots. On ye roots. I've talked about this thing before now. This one you sent recently, advances in plant lipid biochemistry. I've talked about it now. What did you say is the problem? On HU, what do you say is the problem? I've talked about it. I say your slide is well organized. Learn about lipid. Very well. Chisom, I'm still waiting for your slide. Chisom, I'm waiting for your slide. You see what I'm waiting for? I will resend it now. I will send it. If it's the Chisom I'm talking to on YouTube now that is sending this message, please, I'll resend it. But it's my phone I used to use to resend it now. It's my phone I'm using for the live stream now, so I cannot minimize and resend. So just hold on, I'll resend it. I don't know the cost, but I'll resend it. Just hold on. Don't panic. You'll get it. You'll get it. Just calm down. Kadilo Barry, you said I didn't do what? Kadilo Barry, you said I didn't do what? I didn't do what? I'm waiting for you. Anybody remaining? Is there anybody remaining? I want to go offline now. I'm tired. Anybody remaining? Those of you still online, is there anybody I've not attended to? I want to go offline now. I'm tired. I'm tired. Let me give you 3 minutes. Kadilo Barris, I didn't give you what? Kadilo Barris, I'm back to your slide now. Say I didn't give you questions. You that was admiring your slide said you did a nice work. First thing, rename your slide to your math number. Rename that your slide to your math number. Let toxicity and this molecular pathway, lead toxicity. Lead is a heavy metal. So this lead toxicity, lead can interfere with hemoglobin synthesis, hem synthesis. So check how lead can interfere with that. Just check the toxicity. The topic is a straightforward one. How lead toxicity can affect humans. Check that. So in your slide here, I'm saying sources of lead exposure. That's a good one. Is one way they would have asked you. Molecular mechanisms of lead toxicity. Master that. Then this is your slide number 7. All those your number 1, enzyme inhibition, number 2, oxidative stress. Make them bold. Make them bold. Chisom Egewusi, send me a message on WhatsApp. You've said this thing so many times and I don't know the particular Chisom I'm talking to. Chisom Egewusi, that name I'm seeing now. Please send me a message on WhatsApp. I don't understand. I don't know the particular one, the particular Chisom I'm talking to. So, well, that's that. Your topic is good. Your topic is okay. Gifted treasure. I'm just seeing your work now. Gifted treasure. Onyi, I've talked about that thing now. Okay, so you need question. Advances in plant lipid biochemistry, implications, and I told you to read about lipids. Then what is biofuel? Steps in the production of biofuel. So even if it's not strictly part of your topic, they can ask you outside. So what are the steps in production of biofuel? So read about that. Then this person here, your work is in PDF, not in PPT. This person, Ukoka Seno Uwem, convert it to PDF, to PPT, not PDF. Then your slide is interactive, so it's good. I like it that way. But there's no reference, so work on your reference. Then your topic is biochemistry of heavy metals, toxicity and detoxification. I've talked about heavy metals here before. So that settles it. What have I not talked about now? No, Danny and Mandy, if you're online now, they won't ask you any question about how the referees worked and that. Then about your slide 7, calm down after this live stream, please. After this live stream, please. Chisom is now you're coming now. You should have made your points clear so that I will know. Now I understand what you're saying. So calm down. Let me open your slide back. Let me open your slide back. Okay, is your own slide that is not opening on my system, Abby? About this diagram. This diagram here, this diagram you're seeing here. This diagram you're seeing here is the diagram of ATPs. How will I put it now? That round something there is showing how. But I don't know if you're in class when I talked about oxidative phosphorylation, but that whole stuff you're seeing there, that circle, and two leg, that's the cell membrane. Then that whole apartment you're seeing there, that is ATP synthase, enzyme involved in the synthesis of ATP. So, and ATPs are enzymes that is working in that compartment. So that diagram is just showing what ATP is doing. So you're not actually pointing out, you're not actually explaining much about that. If they ask you anything there, tell them that is the ATPase complex. That is how it looks like. Do you understand? I'm sorry that I'm not spending too much time because of how it is now. It's me. Then Chisom Egusi. Now you're coming now. It's now. You'd have told me since now. Chisom, I've talked about this one. I've talked about your slide before now. Jesus. Said make it landscape. So my eyes are red as if I smoke something. In the next 5 minutes I should call it a day. I've talked about this before, Chisom. You're stressing me. Make it landscape. Then master your slide. Your slide is okay. I can't remember the other comments I made about your slide. I said remove a toy in your reference. Then that's that. Jesus, it's past 11. Uchechi, what do you say? John Jewin, you need questions. Where is your slide? You need question. Where is your slide? You need question. Where is your slide now? Chemistry and biology of collagen. Chemistry and biology of collagen. I said you should read everything about collagen, the function of collagen, what collagen is doing in the body. That your topic is actually broad because you are looking at the chemistry and the biological aspect of collagen. So read everything you can about collagen. Your report talks more about collagen, the structure of collagen, the function of collagen. So explain. They can ask you what is the role of collagen in the body, what is collagen doing in the body, how is the structure of collagen, where is collagen synthesized. Collagen is synthesized in the fibroblast. So all those things you talk about it. At this point, I know I might not have satisfied everybody as they wanted, but I feel I tried because there are just so many. I feel I tried. Hey, slides are still coming on. Let me attend to this person lastly. This person, increase Onyeka. Gene polymorphism and folic acid requirement. Please change that background. The background is covering everything now. You would have seen it now. The background on your cover page is covering all the right up. It's not even showing well. So please change that too. Then use bullet points. Use bullet points. Use bullet points. Use bullet points. That is bullet points. Don't just write it as word plain. Give it points. Justify your work. Your work is not justified. Your references are not well arranged, and most of them are outdated references. Like one is 1995, so please update it. Aside that, your work is okay. Gene polymorphism. Polymorphism is actually variation between normally healthy individuals. So you talk about gene polymorphism, how genes varies, variation in genes. Learn about it very well. Most of those lecturers, they are good in those areas. So I've just talked about it. Any other thing? Possible questions again, Chisum. I think the stuff here is just all of you just read your slides in details and do extra research. You see how my eyes are now red and swollen, my headache has increased. At this point, I might not have satisfied all of you very well, but I've tried more than 3 hours. So thanks for joining. If you've not subscribed, click on the subscribe button. If you have any question that is absolutely necessary, send it to my DM. But I'm not responding to any question this night. Send it to my DM. That is if it's absolutely necessary. Please reduce my work. It's just much on me. Please. And I wish you success in your defense. Thank you for watching. Subscribe, please. Thank you for watching. Bye. Let me leave it on for 2 minutes. So in those 2 minutes, if I try, drop something in the comment section. If I did not try to, drop something. At this point, I'm done. So just let me leave it on for like 2 minutes. Say anything you want to say before I finally say goodbye. The 10 persons are still here. 10 persons are still here. If you are here, I'm done, sir. I'm done, sir. All right, bye, everyone. See you next time.